Okay, good morning and welcome to the Day of Science 2021 of the TMS. My name is Anna Teti, I'm Professor of Histology at University of L'Aquila and Chair of the TMS. First of all, I would like to thank the organizers for putting together such an outstanding program. Sara Battista, Giovanni De Gasperis, Antonio Maurizi, Marco Ponzetti, Francesco Salamida and Denise Boncioli. I would like also to thank the sponsor of this event, the Aurogene. They provided the support for 3 awards. So at the end of the day, we will present the Best Presenter Award, the Best Elevator Pitch Award, and the Best Poster Award. So this is the last event of this academic year, and I mean, we organized a lot of events that were really well attended and successful. So we are very happy of this outcome, and of course we are ready to start again at the beginning of the next academic year. So we will have our first TMS in September, on the 6th of September. Everyone here in the audience can contribute. So if you have any a topic that you want to cover, any speaker that you want to invite, or you can also talk yourself. The TMS is interdisciplinary, so no matter what is the discipline of your field of research, just ask and we will organize the event for you. So I think that we can start the Day of Science today, and I will hand over to Luisa Giansanti, who will chair the keynote lecture presented by the Chemistry Nobel Laureate, Professor Jean-Pierre Sauvage. Luisa?
Good morning, everybody. It is a great pleasure for me to introduce Professor Jean-Pierre Sauvage. Since the beginning of the 80s, Professor Sauvage and his research group have been interested in various fields, including coordination photochemistry and solar energy conversion, carbon dioxide electrocatalytic reduction, chemical topology, multifunctional ruthenium and enriching complexes for light photosynthetic separation, multifunctional Parkinson's model of the photosynthetic reaction center, as well molecular switches and molecular machine prototypes such as swinging catenane, master, or compressor. He was a university professor and director of research at CNRS, and since 2009 he is a professor emeritus at the University of Strasbourg. He is co-author of more than 500 publications. He held more than 700 lectures and seminars at international meetings in university and industrial governmental research center. He is member of the Academy of Sciences of many countries, and he received many awards and honors. In particular, he was awarded with Nobel Prize in Chemistry for the design and synthesis of molecular machines in 2016 with Professor Stoddard and Professor Feringa. Today, he will hold a keynote lecture entitled Molecular Machine and Motors, from Biology to Chemistry, on catenane and rhotaxane, family of molecules composed of interlocking and treated links, respectively. So, please, Professor Sauvage, when you want.
Shall I start? Yes?
Yes.
Luisa. Good. So let me share my screen. Do you see my screen?
Yes, we see it. Thank you.
Okay, great. Very good. So, well, let me thank you, Luisa, and the Università degli Studi dell'Aquila, and the TMS organization. I'm very happy to be able to give a lecture this morning. It's very nice. Although I'm in vacation, in the north of Italy, but I'm always connected, connected to the internet. So we will talk about molecular machines and molecular motors, and go from biology to chemistry. Let me introduce you to the field of molecular machines. And I think that everything started with catenanes and rotaxanes. So catenanes and rotaxanes are very exotic species, like exotic species that the chemists like to prepare and to study. And a catenane, in particular, is an interesting species. It consists of several interlocking rings, and for two catenanes, it means two interlocking rings. And such molecules have fascinated chemists for more than a century. Believe it or not, but it's really true. More than a century ago, the people were discussing possible approaches to the making of catenanes, but of course, nothing happened in terms of experimental work. So these compounds have been, of course, at the center of many, many discussions, but in terms of real achievements, it has been very, very limited. I think that the most important contribution from the past is due to Professor Schill. This is Professor Schill, a picture taken in 1984. Professor Schill was working in Germany at the University of Freiburg, and he was the first to propose a very convincing synthesis of catenanes. So he published a paper in 1964, which described the synthesis of a catenane, a two-catenane. Beautiful piece of work, extremely important in terms of history of the field, but there was a weak point, and the weak point was that the synthesis required about 23 individual steps. So very, very long, very tedious. And thus, the people were kind of discouraged by the difficulty of making catenanes, and the field became dormant for several decades. And so we had to wait till the 80s for something really new to happen. And now I would like to explain you how we started in this field. As Louisa said, so of course we have been working in various fields. I am old, and so we had time to explore several exciting research fields. And when I started my group, which was at the very beginning of the 80s, I was much interested in a big project, a grand project, which was the photochemical cleavage of water to dihydrogen and dioxygen, so using solar energy. And it was, of course, very important, very trendy also, a bit after the first energy crisis in 1973, 1974. And there was a lot of money for people working in this field, water splitting. And water splitting, which is still a big problem, still very, very difficult, was based at the time on ruthenium complexes. And in particular, there is a wonderful complex, ruthenium trisbiopyridine, which has great properties. It has a long-lived excited state, photoexcited state. It is highly colored. And the excited state, which is a metal to ligand charge transfer excited state, is at the same time a very strong accident and a very strong electron donor. So this may sound a little bit paradoxical, but it's a fact. And so this complex seemed to have all the properties needed for doing water splitting, for oxidizing water to O2 and reducing water to H2. But it was not so simple, as you know. And there was a problem to us with ruthenium, because ruthenium, it's a very expensive metal. It's a second row transition metal, just below iron. So very rare, very expensive. And we thought, with a good friend whom I'm going to refer to in a minute, that it would be nice to be able to replace ruthenium for another metal. And in particular, we started a big project in collaboration with a photophysicist, an American photophysicist, David McMillan, which was to try and replace ruthenium by copper. And of course, copper is basically free. So that was the project. Sorry. That was the project. And incidentally, David McMillan visited Strasbourg on sabbatical leave. And from the beginning of his stay in Strasbourg, we started to interact very, very strongly, very regularly. And at the time, we had made a molecule, which seems to be today a very simple molecule. But during the 80s, at the beginning of the 80s, it was new. It was a new molecule. And we had developed a synthesis for making this compound, in fact, for other purposes, more related to transition metal-based catalysis. And when Dave saw the molecule, he jumped around and he said, well, we must collaborate. You will make the copper complex of this compound, and we will study all the photochemical properties, do all the photophysics, and it will be a nice collaboration. And it turned out to be the case. It was a very nice collaboration, completely photochemistry oriented. And we found, thanks to David McMillan and his group, that this complex had properties which were very close to those of ruthenium-3 biopyridine. So it was a great success in terms of photochemistry and photocatalysis. That was the beginning. But there was something else, probably more important. And I will now explain you something else. If you look at the drawing here, you can materialize the endpoints of the two organic fragments. So a blue dot here, another blue dot for the two ends of this ligand. You can do the same for the other ligands, red dot here, another red dot for the other endpoints of this other components. And now, it's very simple. If you interconnect the two blue points and separately interconnect the two red points, what are you obtaining? It's pretty obvious. You make a catenane. You make a ring here, incorporating the red fragment, another ring here, which incorporates the blue fragment, and those two rings are interlocking with one another. They are married forever. And so we had this very simple idea, and we thought, well, we are not very familiar with catenanes. The field was not very active anymore. It was not even active at all anymore. And so we had a long discussion, my group, to know whether we should jump in a new field, in a virgin field, more or less, or continue to do what we were doing, feeling very comfortable with catalysis, photochemistry, electron transfer. And we decided to jump. And so we jumped, and thanks to my good friend, Christian Buscheke, who was a fantastic organic chemist, we started the project, and within about 8 to 10 months, we had our first paper. So as I said, she was a fantastic organic chemist, and she tackled the catenane problem with a lot of enthusiasm and fantastic skill. And we published in 1983 a paper. We published it in French, mostly for fun, because we were a bit tired to have to read English all the time. And we published in a, let's say, moderately good journal, which was Tetrahedral Letters. And that was the very beginning. And I should say that from this moment, catenanes appeared as accessible species. It changed completely the view that chemists had on catenanes. From very exotic and impossible to make species, they became all of a sudden very accessible species. So the first strategy, which we call the entwining strategy, is quite simple. We start from the molecule, which is represented here in the corner, and we mix it with copper. So let's do that. Copper 1, we generate this complex, which is the entwine complex. Two ligands are entwined within one another. And then we cyclize on the right, in the same pot, we cyclize on the left, and we have our catenane. 3 steps. And the second strategy is a little bit more complex, but not terribly complex. We have to prepare in advance a ring which incorporates the chelating fragments, which is a phenanthroline-based fragment. We will mix it with copper. Separately, we will add an open chain chelate, and finally, we will cyclize. Copper, open chain chelate, and we cyclize. And it's a slightly longer route, maybe 5 steps or so, but it works equally well. And so just to summarize, we could make a catenane, copper-containing catenane in 3 steps. We could demetallate using potassium cyanide. This is very easy, quantitative. We generate the new species, which is the metal-free catenane, and we can go back by adding copper. So these 2 reactions are quantitative and instantaneous. We were fortunate to be able to crystallize the species and to have X-ray structures of them. So you see here a relatively globular species, quite compact, and here a very open structure from which the 2 rings can glide more or less freely within one another. So we call this conversion here between the copper complex and the metal-free species and vice versa, metamorphosis. And this is indeed a metamorphosis because you cannot recognize the molecule anymore, starting from one state to another. So that was the very beginning. And now I will jump years and years and years, and we became mostly interested in topology. Topology is a very complex concept, which I'm not going to discuss, but we have devoted a lot of time, a lot of effort on making new topologies. And in particular, there are a few examples here. A 3-catenane, probably the most exciting and the most difficult, a knotted ring. This is a single closed curve, but it has been knotted. And this species is really fascinating. Another example is the doubly interlocking catenane, which some people call the Solomon rings, especially topologists, mathematicians. And so this, in a way, was the start of a new field, and many groups have been interested in molecular topology since the very beginning with our contributions. Now let me pay homage to the people who work in the field of catenanes. So this is, of course, as I say here, a bit of history. Ed Wasserman, an American scientist, mostly a chemist, but who was crazy about chemical topology, was working in a big chemical company, DuPont de Nemours, and he proposed some kind of theory and also a statistical approach to the making of catenanes. But honestly, it was brilliant in terms of intellectual approach, but not very practical. Professor Schill and his colleague, Professor Lüttinghaus, I already mentioned their fantastic contribution. our team, and later on some other pioneers, Fraser Stoddart and his group, Chris Hunter, Fritz Vöckle, Makoto Fujita in Japan. And I think from the mid-90s, the field really blew up. It developed very rapidly thanks to the contributions of really highly creative research teams. Now, let's move on and talk about molecular machines. I have two statements here which are very important. The first statement is that chemists, most of the time, consider their molecules as static objects. Molecules which, of course, can move, can distort, can elongate, can vibrate, but for which we have no interest in terms of controlled movements, controlled motions. So there are stochastic movements, but that's all. Biology is totally different. And in a way, biology played a very important role for us because it was to a large extent a source of inspiration. In biology, controlled motion is absolutely essential, and you find it everywhere. So there are rotary motors, linear motors, there are walkers, molecules which walk on path, systems undergoing contraction or extensions, compressors, etc. And most of these biological molecular machines belong to the very important family of molecules which are the motor proteins. Motor proteins are everywhere. I will just show you two examples, which maybe some of you are very familiar with. I'm sorry for that, if it is the case. We will talk about ATPase or ATP synthase when it's making ATP. ATPase is a rotary motor, which is present in everything which lives on Earth. From the most primitive organisms to mammals, energy is based on ATP, and it is based on ATP synthase or ATPase to make ATP or to hydrolyze ATP. And the second example will be the walker, which I was mentioning. So you can find some beautiful videos on the net of ATPase, and I would like to insist they are not science fiction videos. Some of them are science fiction, but the most serious videos are purely scientific. They are based on scientific data, X-ray studies, hundreds, if not thousands of studies by molecular biologists. And now it is absolutely clear, it is accepted by everybody, that ATPase is a rotary motor. It is embedded in the bilayer membrane of mitochondria. You know that mitochondria are the energy factories of the cell, and it can rotate very, very fast. So you have a shaft here, an axis, which is threaded in the cavity of a big cluster of proteins, 6 proteins. And in between those proteins, between the red and the green, you have some voids, some small cavities in which ADP, which is yellow, and inorganic phosphate, which is blue, can insert themselves. And then there is some kind of mechanical process which forces ADP and inorganic phosphate to react. And you generate ATP, and of course, ATP is expelled as the purple species. It can rotate very fast. I mean, when you have a lot of ATP and you want to hydrolyze ATP, this is not in the cell. I mean, it can be done separately. It can rotate as fast as a Formula 1 engine, so 10,000 rounds per minute. The other extremely impressive molecular machine of biology is that of Kinesin. Kinesin is in charge of several things, but the most important thing in the cell is that kinesin has to carry molecular system from one place in the cell to another place, where the cell needs the molecules that have been fabricated in the biosynthetic factories. And so the kinesin is this tiny thing. It has two heads. This is what our biologists would say. I think we chemists would consider that they are rather 2 feet, because this beast here walks on the microtubule, so it's a big tube here. And at the back, you have a big bag full of molecules, proteins, nucleotides, nucleosides. And all these molecules are carried from one dot in the cell to another dot, very far away. It can be in the opposite place of the cell. And this goes very, very fast. Of course, it looks extremely slow on the video, but I think the kinesin on its microtubule can cross the cell completely within a microsecond or so. Now let's look at what chemists have done in the field of molecular machines. And we will go back to ketones and vitaxins, because this is how it started. Why are ketones and vitaxins so promising in terms of motion? It's very clear. If you look here, you have a very simple vitaxin. A ring can glide from a position on the left, let's say, to a position on the right. And if you can control the motion, you are very close to making a linear motor, like a piston moving in a cylinder. Similarly, here, if you can rotate or pirouette an axis within a ring, or here, for a ketone, if you can move one ring within the other ring, considered as fixed, you are on your way to making rotary machines, rotary motors. So clearly, ketones and vitaxins will play a very important role in the field of molecular. So let me explain you how we started. So that was many years after we made the first ketone. It was in 1994. And the principle is very simple, you will see. So we start from a ketone, which is also easy to make. And this ketone is slightly more complex than the one we have seen before. We have introduced at the back of the system a third pyridine, 1, 2, 3 pyridines with 1, 2, 3 nitrogen atoms. And these nitrogen atoms might be able to interact with the transition metal. Now let's start from here. We have copper 1 plus, and copper 1, it's a D10 transition metal. It is very happy when it is surrounded by 4 nitrogen atoms. So here we have a very happy copper 1. In other words, we have a very stable copper 1 complex. Now let's send a perturbation to the system. Let's oxidize copper 1 to copper 2, abstracting an electron. We generate now a copper 2 center, which is still 4 coordinated, but copper 2 hates to be tetrahedrally coordinated. It prefers to be 5 or 6 coordinated, and so the system will react. We have here 3 nitrogen atoms, and these 3 nitrogen atoms will replace the 2 nitrogen atoms which were originally interacting with copper 1. So the ring will rotate here, it will undergo a half turn, and after that you generate a very stable complex with a very happy copper 2. 1, 2, 3, 4, 5 nitrogen atoms are participating in stabilizing the copper 2 center. You can, of course, go back you reduce back to copper 1. Again, you have a pirouetting motion, and you regenerate the starting form of the copper catenate. So this is what we did, and it was not perfect. We had no control over the directionality of the motion, and on top of that, it was very slow. It used to take seconds to minutes from the system to move. So we have a nice video, or at least I find it nice, which shows that we can go from copper 2 here to copper 1. This will lead to a complete rearrangement. You can again oxidize copper 1 to copper 2, etc. And you can do that as many times as you like in your computer. But the beauty of the system is that you can also do that as many times as you like with the molecules. They are perfectly stable. Just to tell you, we were not so happy at the beginning because the system was very sluggish, very slow to react. And so we spent a lot of time, more than 10 years, trying to generalize the concept and also trying to improve the rate, to increase the rate. And so we, after, as I said, a couple of years, we could make a Vitaxins, which are in a way more exciting in terms of potential applications. They can move within microseconds to milliseconds. So that was for our work. Now let me show you some very important pieces of work done by other people, and in particular, our good friend Fraser Stoddart and his group did beautiful work on molecular shuttles. So this is a shuttle because a ring can shuttle between two positions, a position on the left of the axis and another position on the right. So here are the molecules they made. You have a blue ring here, which is a strong electron acceptor, highly electron withdrawing species, which can interact with a green station or a red station. And this is governed by electron transfer. You can trigger the motion by oxidizing the green station and thus killing the attractive interaction between the blue ring and the green station, or reducing back the oxidized green station to the neutral form of it. So that was very nice. It was published the same year as our swinging catenate, as we called it. And as far as I know, I think it can be considered as one of the very, very first molecular machines. That found some very important implications in another field, and Fraser Stoddart and Jean-Pierre have devoted a lot of time on trying to move towards, let's say, molecular computing or electronic memory devices based on molecules. And if you are interested in this field, I invite you to read this relatively old paper on how to store information using molecules. I think it's a fantastic piece of work. Let me now pay homage to another very, very good friend, Ben Feringa, and his co-workers. And Ben Feringa published a completely revolutionary paper in 1999. It was a rotary motor. For the first time, chemists were able to make a rotary motor, a molecule which behaves as a rotary motor with perfect control of directionality. And this is certainly the most difficult. So this drawing is very nice. It has been prepared by Alberto Credi and Vincenzo Balzani in Bologna. It's more, let's say, more beautiful than the original drawing prepared by Feringa in his book. You have a double bond here, and as you probably know, if you have a highly hindered double bond, you can photochemically isomerize it. So you shine light on it here, you trigger a photoisomerization. And so you convert this to that. And then what was really beautiful is that if you are patient enough, the motion goes up, and this fragment here, which was here, will go up the motion, and they realize that. And then you can, again, shine light, so you have a second photochemical step. The system will continue to rotate, and you generate the third form, and the third state of the molecule, and finally you heat it, and you regenerate the starting form. And this is a complete cycle. You have completely rotated the molecule. They published a very nice video on the web. I hope you can visualize it. So you have the double bond here, and you will have 4 steps. Photochemical step, then a thermal step, third, a photochemical step, and fourth, the final thermal step, which will bring you back to the starting state of the molecule. So let's hope that you can visualize it. You shine light, you heat, you shine light again, you heat, you shine light, you heat, you shine light, you heat, etc. Well, I hope you can visualize the video. I think it's not so simple, and I'm sure that for some people it will be very, very difficult, but I cannot help you. If you cannot visualize, I just apologize. There is nothing I can do to help you. Now, let's move on, and let me talk about some work which we did in our work, which started about 20 years ago. We were very much interested in artificial muscles. So that would be a fantastic machine, a system which you can contract or extend at will by sending a signal to the molecule. And our work, I mean, it's not very nice to say that, but in a way it triggered some interest for molecular muscles, and several groups have been interested in artificial muscles based on various molecular systems, and beautiful work. Just to remind you, the way biological muscles, our muscles, they work by gliding filaments. So you have the thin filaments in red here. It's a polymer, an actin polymer, and those thin filaments can glide along thick filaments, which are myosin elements. And myosin has an ATP hydrolysis action. And so when you contract the two fragments which are here, the red set of thin filaments, which move towards one another, and you generate this contracted form. You consume ATP, and when you relax, of course, you do not consume anything, as you know. And so we made a relatively complex rotaxane. It's a rotaxane dimer of this type. We have a pale blue fragment. We have an identical fragment, which is the dark blue one, shown here. And I have to apologize because the system is really complex, and it would take us at least 10 minutes to explain in detail how it works. So I just show you the principle. The principle is that we can send a signal to the molecule, and it will contract, go from a length of 8 nanometers to an overall length of 6 nanometers. And we can send another signal, a chemical signal, which will go back. So we can do that several times, contract or elongate, and And as I said, it has been generalized, and several groups made the system much more efficient. And in particular, some groups prepared fibers, very tiny fibers, and they can contract them or elongate them by just changing the pH. We were interested in trying to make a big receptor and thus contract molecules. And this is a very complex system, but I think we can still have time to discuss it, not too much in detail, but I will show you the principle at least. So the project was relatively simple. We wanted to make a 4-rotaxane consisting of 4 covalently independent fragments, 2 dumbbells. So these are the 2 dumbbells here, and 2 bis macrocycles. 1 ring here, 1 ring here. So this is a bis macrocycle, the same macrocycle here. And we will incorporate 2 plates, 2 square-shaped plates at the center. And the project was first to make such molecules because multi-rotaxanes were not so common at the time. And we thought it was also a synthetic challenge, but also to be able to incorporate a guest, an inviting molecule, inserted between the plates, and then at some stage, bring the plates to close proximity and contract the guest. That was the project. Relatively simple. So this is the molecule. Well, I'm very sorry for the people who are scared of complex molecules. I know it is relatively complex, but it is highly symmetrical, which made it relatively accessible in terms of synthesis. So again, we have the 2 dumbbells here and here, very big stoppers to prevent the system from unthreading, the 2 bis macrocycles, and at the center, the plates are relatively simple. They are porphyrins, zinc porphyrins. And you know that zinc porphyrins are relatively easy to work with. They are very robust, very rigid. So they are ideally the plates we would like to use for compressing something. We will incorporate a guest and see whether we can compress it. So something which was unexpected and which came as the icing on the cake, we could obtain an X-ray structure, and this is the X-ray structure. And in fact, it doesn't show anything completely new. This is exactly the structure we had expected with the dumbbells. You see the dumbbells again here. The 2 zinc porphyrins here and here. And there is some space in between those 2 porphyrins in which we can incorporate small molecules. So Darco, 4,4' biopyridine, and related small molecules. And now, so it worked fine. And now let me show you what happens. So this is a video which is based on X-ray structures, not on science fiction again. We have the 4 copper ions, and it will be quite simple. We will send a chemical signal, very small amount of cyanide, and the copper ions will be removed, and the system will collapse. So for the moment, we have to think that we have a small molecule in between the 2 porphyrins. This is just to remind you the shape, the general shape of the molecule. We send a signal, and the system collapses completely. This has been fully demonstrated. And the molecule which was inserted in between the porphyrins is first compressed, compressed, and compressed, and finally, of course, expelled. We can go back we had a little bit of copper, we regenerate exactly the same structure, which is able to complex small molecules in between the two profiles. So let me finish up by telling you that the field of molecular machines is nowadays very active. There are probably hundreds of groups working in this field. There were several examples originally. Peer-weighting catenanes, so that was our work, the work of Fraser Stoddart and his book Molecular Shuttles. Rotary motors, let's say photochemically driven rotary motors, the work of Ben Ferringa, molecular muscles. Several groups have been working in this field. Compressors, molecules which can walk on a rail, and this is remarkable. This is in particular the work of David Lee in the UK and his co-workers. Another very impressive piece of work is that of molecules mimicking the behavior of ribosomes, so able to synthesize small peptides in a perfectly controlled fashion. Again, the work of David Lee and his group, and some others. So we have no time to discuss in detail what has been done. Let me say that before the work on molecular machines, chemists were not really considering their molecules as, let's say, having motions which can be controlled, apart from a few exceptions. And so I believe that the work of the people working in this field have completely changed the way that molecular chemists look at their molecules nowadays. They know that they can make molecular machines and molecular motors. Applications, what can we think of? This is, of course, very difficult to talk about applications, but there are many fields in which we can hope to find applications. For the moment, there is only one commercial application which is related to polymers or films which can self-repair because the molecules can move and regenerate, let's say, the exact shape of the screen. But probably the most important application, and there are many, many people working on that, is trying to vectorize drugs, biologically active species in biological media, in blood, let's say, so as to transport and deliver therapeutic molecules exactly where they are needed. So targeting tumor cells or even other systems, viruses or bacteria. I would say, that it is very important to keep in mind that basic research is at the origin of everything which is technologically important today and which are parts of our everyday life. At some stage, it started with the basic science. Let me thank the people who have been doing the work. I mean, I had a wonderful team of people, and of course, it is their work. So the beauty of the French system, which is a little bit like the Italian system, so you have the CNR, we have the CNRS. Although the CNRS people are more integrated within the universities than the CNR people, who most of the time work in separate research centers. But many of these people were CNRS researchers, and some others were university professors, so they have done great work. There were many young students, PhD students, postdocs, who also contributed enormously to to our work on topology, in this case, or on molecular machines. I'm very sorry that we have no time to name them, or at least not to name them all. The very last system I spoke about, the compressor, this is the work of these people. So some PhD students and postdocs in black and permanent people in blue, Jean-Paul Collin, Valérie Heitz, two fantastic colleagues, and younger permanent people. And finally, let me thank again my team, the French CNRS, my university, Strasbourg, my second university, Northwestern University, where I spent 3 years after the CNRS suggested me to retire. I was not ready to retire, so I had to move somewhere else. The European communities, my new institute, ISIS, where I have been invited to join, thanks to Jean-Marie Lane, my former mentor and very good friend, Malcolm Green, a great organometallic chemist, passed away recently, very unfortunately. Two fantastic teachers, my family, and my two good friends, Fraser Stoddard and Ben Ferreira. So I hope I haven't been too talkative, and I would like now to stop at this stage. Thank you.
Thank you, Professor Sauvage, for your interesting lecture, real excursus on molecular machine and made by chemists and also a connection with the biological molecular machine. I think that, as you said, this system, you said the beauty of these systems. I agree with you. I think that it's really fascinating what we as chemists, you and the researcher, did to control the motion of molecules. It's really complex, and you explained us very clearly how you get to these results and the different kinds of molecular machines that are described in literature. So I think that in your lecture, it was clear also the importance of the collaboration with other groups. I mean, join the expertises of many researchers, because to obtain results, I think that it's important that each researcher put his contribution, no?
Yeah, if I may say, in a way, with Professor Vincenzo Balzani, is a great friend, but there are also other people, Lucia Flamini, who is a CNR researcher. She's now retired. Francesco Barigeletti. I mean, with Bologna, well, I've visited Bologna, I don't know how many times, but many, many times. And I think we have published more than 50 papers together. And so it was a fantastic collaboration, with the Bologna people, but also some other people, of course, in the US. It is very, very important to be kind of delocalized and to have very good contact, scientific contact, but also friendship with as many researchers as possible.
We have a question from the audience.
Shall I stop sharing my screen, or?
Yes, you can.
Yeah.
The question is from Michela Ciocca. Can we say that xenobots are molecular machines, even if they are made by cells? What do you think about this new technology?
What? I'm sorry. Cellular?
Xenobots. The question is on the chat if you want to read it.
Yeah, okay.
On the right.
Yeah. It's a long question, though. No.
Can we say in the lower part?
The lower part.
Can we say that xenobots are molecular machines even if they are made by cells? What do you think about this new technology?
I try to find a question. I have a question by Giulia Tatangelo.
Now you have to...
So who is the person who is asking the question?
You have to arrive at the end of the chapter.
Yeah, you're right. There are several questions by various people here.
So another question, naive question.
Oh yeah, xenobots. Okay, yeah. I would say yes. I would say yeah, sure. I mean, basically any biological system is an, let's say, complex molecular machine-based system. Yeah.
Okay, another question. That's a naive question for a biotech. Can you envision incorporating linear molecular machines in biodegradable patches, for example, to substitute stitches when suffering a severe cut or other injuries?
I mean, this question, this is exactly what some people are trying to work on. Clearly, today, I mean, this is still science fiction.
Okay.
But personally, I'm convinced that someday, we will be able to mix artificial molecular machines and biological systems. If I may say, for the moment, the big problem is to have biocompatible molecular machine systems.
Okay.
Biocompatibility is a big problem.
Of course, to use it in this field. So, if there are no other questions, I thank you for. I really thank you for your lecture, for participating to our congress today. I hope that I am sure that the students appreciated your clear and interesting lecture, and I hope that you can stay with us and follow the other lecture, the other talk of the students, if you want, obviously. And so, thank you again, merci. And we.
Thank you, Lisa. Thank you.
There is another question, Luisa.
Sorry?
There is another question in the chat.
Sorry. There is another question. I didn't see it. What kind of large-scale industrial development do you envisage for molecular machinery?
Yeah, I see the question. Large scale, I'm not sure what it means, but in terms of drug delivery, if one day it works, if drug delivery systems work in the body, clearly they will be used, by lots of people. But still, as I said before, there is only one type of application, only one, in the field of materials, and there are no real applications oriented towards medicine or nanomedicine. We have to be patient for that.
Sure, obviously. So now I think that there are no other questions, if I'm not wrong. So thank you again, Professor Sauvage. Now there will be a break of 15 minutes, during which some elevator speeches will be projected to give the opportunity to the young participants to the Congress to introduce themselves and their research.
Thank you.
Thank you again.
Just a few seconds, and then we will project the elevator pitches. We will also have a votation, so you can vote the one you prefer. So just bear with us for a few seconds. I'm going to do that. So I'm going to share my screen. Can you please let me know if you can see the screen?
Hi everyone, I'm Arianna Sinibaldi, PhD candidate under supervision of Professor Armando Carlone in L'Aquila University. In this work, I studied the increasing of reactivity by using supramolecular catalyst in aqueous solvent. Just only the first heterochiral peptidic sequence is able to pre-organize itself in a supramolecular fibrils, but not the other one sequence. So, after the observation by using confocal microscope and TEM, we tried the catalytic system in a benchmark reaction. But now I will show you how the system works in a macroscopic version. The single peptidic sequence is able to self-assemble in more organized structure by forming the catalyst, the fibrils. After that, the first reagent interacts with the catalyst and it is activated via an amine. In this way, the intermediate could attack the second reagent by forming the desired product. If you are curious, check my poster and see you on TMS Day of Science. Hi everyone.
Okay, that was the first from Arianna Sinibaldi. There goes the second from Francesca Di Donato.
I'm Francesca Di Donato, so nice to meet you. I graduated from the University of L'Aquila and I am actually a PhD student in the same university. I am carrying out my research in the field of development and validation of analytical methods for food traceability. I know it's a big, complex concept, that's right, and it also has deep economic and environmental implications. For example, we have recently evaluated the potential of the aroma profile in the characterization of 3 typical Italian pecorino cheeses with the aim of verifying the truthfulness of what is on the label, and we have developed an effective tool to do this. If you are interested in details, just take a look at my contribution to the TMS Day of Science, titled Edge Space Solid Phase Microextraction coupled with gas chromatography, mass spectrometry, volatile fraction determination, and chemometrics for the discrimination of typical Italian pecorino cheeses. You know, the academic world is trying to help, looking for efficient indicators of quality and authenticity, but all is for nothing if the full cooperation with governments, producers, and consumers is missing. And also an effective legislative contribution will be needed. Maybe you can help us. Hoping to see you soon at the TMS Day of Science.
Okay, now the third elevator pitch from Valeria Nori.
Hey everybody, I am Valeria Nori and I'm currently a PhD student in organic chemistry in L'Aquila University. I'm working under the supervision of Professor Armando Carlone. The main focus of my research is studying and exploiting different catalytic strategies in order to synthesize pharmacologically active molecules. But what is a catalyst? A catalyst is something such as small organic molecules, lights, or metals that can speed up chemical reactions. In this project, we used organocatalysis in order to perform a Michael addition of masked acetaldehyde to nitroalkenes. Thanks to this strategy, we set up a very simple, safe, and cost-efficient protocol. But what's the real challenge here? The real challenge is using acetaldehyde, which is really toxic, highly reactive. In fact, it can undergo self-condensation reactions really easily, and it has a really low boiling point. How did we figure out our problem? So, do you know the Farmer dilemma? A farmer went to a market and bought a wolf, chicken, and wheat. On his way home, he has to cross a river, and he could just take only one of his purchases with him. If left unattended, the wolf would eat the chicken would eat the wheat. So, how did the farmer cross the river? Sometimes we, organic chemistry, are required to think as a farmer of the story, adding some reactive partners to the other ones. This is the case of acetaldehyde, which is masked as acetaldehyde dimethyl acetal. If you are curious and you want to know more, check out my poster and see you on TMS Day of Science.
Okay, now the next is from, let's go with Claudia Marconi, so we can finish the chemistry ones.
What happens if lysozyme is treated with hypochlorite? And why is it important to answer this question? Because both lysozyme, which is an enzyme with antibacterial function, and hypochlorite, which is a powerful oxidant, play a fundamental role in the immune system response to the attack of external pathogens. But in some occasions, such as in chronic infection, an excessive production of hypochlorite can oxidize host protein, including lysozyme. My name is Claudia Marconi. I am a PhD student in material science, and I studied this question in my degree thesis in chemistry at Sapienza University of Rome. In my poster that I will present at the TMS Day of Science at L'Aquila, I show the results of my instrumental investigation. Visible spectroscopy, circular mechanism, dynamic light scattering, fluorescence spectroscopy, X-ray diffraction, atomic force microscopy measurement, show that lysozyme undergoes conformational changes caused by oxidation by hypochlorite, which may explain the loss of its enzymatic function and the aggregation phenomena that I observed in vitro. The mechanism of this aggregation of lysozyme in other proteins is not fully known, but it is a topic of great interest because this aggregation at the infection sites can lead to the formation of amyloid fibrils, which are insoluble deposits found in degenerative disease called amyloidosis, such as Alzheimer's disease, for example.
Okay, now we change topic. Let's go with Michela Ciocca.
We all know that breast cancer is the second leading cause of cancer death in humans. But maybe you don't know that the major problem is the relapse after years of absence of the disease. Are you curious to understand why and how it's happened? I do, and that's why in our bone biopathology laboratory, we are studying how breast cancer cells manage to escape from treatment by hiding for years, then generating a new tumor distant from the primary site. This small population of cancer cells enters the bloodstream and is able to reach the bone, where it comes into contact with a particular type of osteoblasts that are cells that normally produce new bone. Thanks to this binding, breast cancer fall asleep and blocking their cell cycle. They are able both to evade our immune defenses and to mimic the behavior of resident cells of the bone, such as the so-called hematopoietic stem cells. Now that you know this story, you can understand how important it is to study how this happens and what are the mechanisms that allow these dormant cells to survive for years and then awaken, giving rise to a new tumor. I'm Michela Ciocca, PhD student of the Bone Biopathology Lab handled by Professor Anna Tedi. If you want to know more, you can go and read my poster, which explains what we have already understood and what else there is to understand about this process. Hoping to see you soon, I look forward to seeing you at the TMS Day of Science.
Okay, now for the last one from Pier Giorgio Patrizzi.
Hi everybody, I am Pier Giorgio Patrizzi and currently I am a fellow from Bone Biopathology Laboratory at University of L'Aquila and I'm working under the supervision of Professor Anateti. The main focus of my research is to study the rare genetics bone disease, and one of them, which I would like to talk about in this video, is the Cole-Carpenter syndrome. But what is the Cole-Carpenter syndrome? The Cole-Carpenter syndrome is a rare genetic disease that displaying an autosomal dominant inheritance. It affects mainly the bone, which becomes brittle and fractures many times. Genetically, the Cole-Carpenter syndrome is caused by the heterozygous epsilon 393C mutation in the P4HB gene that encodes for the prolyl 4-hydroxylase beta subunit, and to date this unknown disease has no cure. But what is our proposal? The central hypothesis of our proposal is to demonstrate that the first animal model of the Cole-Carpenter syndrome, generated in our laboratory, represents a useful tool to in-depth investigate the mechanism underlying this genetic disorder and to develop therapeutic strategies for the Cole-Carpenter syndrome. If you are curious to know more about our project, we advise you to look our poster and see you on TMS Day of Science.
Okay, so this concludes the projections of the Day of Science elevator pitches. So if you are curious to also watch the posters, I will now send you the link to access the Google Drive folder. Go. You can find that in the chat, or I put a multimedia visualizer down there in the web interface. So now we switch to the next session. We will have the votation later in the next coffee break since we are running a bit late. So I will now give the virtual floor to Denise Bongioli. There she is. Okay, Denise, can you speak?
Yes, can you hear me?
Yes, perfect.
Okay, thanks.
I'm also trying to find Juliana. There she is. Okay, activate audio. All right, thank you and good luck.
Thanks, Marco, and welcome everybody to the physics session. So we change topic from the first lecture to the talk by Dr. Juliana Galati, that I thank a lot for her for accepting our invitation today. So we are going to hear from Juliana about neutrinos, but let me just say a few words about Juliana's activity. She's a researcher in physics at the University of Paris, and for many years she was involved in experimental research in neutrino physics, and I think she's going to tell us something about this elusive and this very particular particle. But actually now she's involved in medical physics, so she's applying the results and tools deriving from research in particle physics to new clinical therapies. She is also very involved in many activities, such as she's an author and speaker for the podcast Scientifica since 2012, and since 2019 she has been an author of the Super Quark Plus program. So so we are very happy to have Juliana here. And so from myself and from all the TMS committee, we thank you a lot, and then we let you start your presentation if you want to share your screen, Juliana.
Yes, thank you for the presentation and for the invitation. So can you see the slides?
Yes, perfectly.
Okay, so good morning to everybody. And today I will tell you a story, a true one. It's the story of naughty particles called neutrinos. Wait a second, because, okay. So sometimes I say that for many years I've worked inside a mountain, where I used a soccer net to try to catch mosquitoes moving at nearly the speed of light. Well, not literally. I'm not crazy, or at least not too crazy. As Denise said, I'm a physicist, and I was not trying to capture literal mosquitoes, but neutrinos, these particles which have been making scientists go crazy for almost 90 years. Let's do first a short summary of particle physics. So in this table, you can see a funny imaginary representation of all elementary particles we know today. Elementary means that you cannot divide it anymore. It's not constituted by something else. Well, there are 12 elementary particles, but everything we see and we touch is made of atoms. And in the atoms, we find only two types of quarks, the up and down quarks, which constitute protons and neutrons in the nucleus, and electrons. Neutrinos are here in the table. There are 3 of them, and as you can see, they are represented as gangsters. Why? Well, everything began as a matter of principle, a matter of conservation principle. In 1896, the radioactivity decays were discovered. It means what's radioactivity is when a nucleus transforms itself, we say it decays into another nucleus, which is a little lighter, and an electron. What's wrong with that? In 1926, physicists were struggling to solve a mystery about these decays, called the beta decay problem. It seemed that both energy and angular momentum were not conserved in radioactive processes. And the universe isn't very happy when energy and momentum aren't conserved, and neither are physicists. So let me explain you how to be a physicist without even going one day to university. So we have a problem, and in this case we had a problem. Numbers don't add up. So on one side of our equation we have like 1 + 1, but on the other side we had 3. So that's something wrong. How to solve this mystery? Well, physicists add an hypothetical invisible number, so that 1 + 1 plus this invisible 1 makes 3, and everyone is happy. So it can seem like a joke, but this was exactly what Pauli, a brilliant physicist, did in 1930. He conceived the existence of a neutral, non-interacting particle just to make ends meet. So he wrote this idea in a letter that is quite funny, and because he didn't want to go to the meeting with all his colleagues because he had a ball, and going to dance is much funny than going to a conference with physicists, boring physicists. And this idea was really crazy, and he was the first that didn't believe in its idea because it was almost impossible to prove it. And indeed he said, "I've done a terrible thing. I invented a particle that cannot be detected." But luckily, experimental physicists are very good, and it took like 25 years to detect the first neutrino, but at the end they proved that this crazy idea was right. And they confirmed the existence of neutrinos, and of course that was worth the Nobel Prize. But then they understood that there were more kind of neutrinos. They discovered also two types of neutrinos, and the last one, the tau neutrino, was discovered just almost 20 years ago. But have you ever dealt with neutrinos? Where do neutrinos come from? Well, for sure you have. At least once in your life, I think that you have been near bananas, for example. And bananas contain a little quantity of potassium, and potassium has a radioactive isotope that produces neutrinos. But not only bananas produce neutrinos. Neutrinos are very important. They are the most common particle in the universe after photons, that are the particles of light. And they are produced in all nuclear reactions. So, for example, in the stars like the sun, in the nucleus of our Earth, by supernovas in other galaxies, by nuclear power plants, and they were produced even during the Big Bang. So neutrinos hold the key to explore the most remote universe and look back in time up to the Big Bang. Indeed, they bring us information otherwise unreachable because they have a unique feature. They can pass through everything. There are billions of them passing through your bodies right now. Of course, you can't feel them because for them, atoms are just big empty spaces. Neutrinos can travel through entire galaxies without eating any other particle. They can blaze through our Earth as it wasn't there. So that's why my job seems so crazy, like trying to catch mosquitoes with a soccer net. Maybe you can't imagine, but you also produce neutrinos, millions of neutrinos per day. So neutrinos are so frequent, so common particles, but we still don't know much about them. Despite all the present big technologically advanced experiments, we still don't know, for example, how much a neutrino weighs. We are not able to measure their mass because it's like trying to weigh a mosquito with a grocery scale. So we are not sensitive enough. Just to give you an idea, if an electron, which after neutrinos is the lightest particle, weighed as much as a bear, then a neutrino would weigh much less than a mosquito. And so for many years, physicists thought that neutrinos were massless, so they didn't have mass at all. And this is not so strange because also photons, the particles of light, don't have mass. But there's another way to understand, at least if they have mass. So neutrino, as we said, come in 3 distinct species. We say different flavors, even though no scientists have ever tested them. And these are electron neutrinos, muon neutrinos, and tau neutrinos, named after the 3 charged particles to which they give birth when they collide with an atom. So if neutrinos have mass, their flavors can oscillate, which means that they can be born of one type and then over huge distances transform into another. And so this is a way to understand if they have mass in an indirect way. Imagine starting a long-distance dog sledding race with a pack of beautiful huskies. And then, crossing the finishing line, you discover that one of your dogs has turned into a cho-cho. Well, this is exactly what happens, but with neutrinos instead of dogs, of course. So why we call this process oscillation? Because mathematically we can describe neutrinos as a combination of waves. In this picture, we see a simple description with just 2 neutrino flavors. Indeed, we know there are 3 of them, but let's just simplify it a little bit. So according to the position of these waves and the wave that they form if we sum them, we can obtain one neutrino or another kind of neutrino. Okay, that's quite complicated, but just follow me. We will see a little movie now. And this double pendulum, it's a neutrino. So the 2 pendulums together make a neutrino. And depending on how the 2 pendulums are one respect to the other, we have one kind of neutrino or another kind of neutrino. So, for example, if they move parallel one to another, we can imagine to have the muon neutrino. If they move in the opposite way, we can imagine to have the tau neutrino. So I show you the movie. So we start with muon neutrino, parallel movements of the 2 pendulums, but at a certain point, we have an opposite movement between the 2, tau neutrinos. But if we wait, we have again muon neutrino, and if we wait another time, we have again tau neutrino. So that's why we call it oscillation. How we can test this idea? Well, to test this hypothesis, we can make an experiment where we take neutrinos, all of the same flavor, muon neutrinos, that were produced at CERN, where there is a big accelerator in Switzerland. And then all these muon neutrinos run underground through the Earth at almost the speed of light, since they arrive at the Opera detector in Italy. Opera was the name of this big experiment that was placed underground in the Gran Sasso laboratories. So what we want to see is that sometimes a muon neutrino transforms into a tau neutrino. So at the end, we see something that was not present before. The problem is that neutrinos are almost undetectable. All tracking devices developed in particle physics can only reveal electrically charged particles as they pass through a medium and they interact. Neutral particles, instead, can be seen indirectly when they interact and they produce charged particles. What's the problem? That neutrinos are neutral, but they hardly interact. So, as I said, they can travel through the entire universe without hitting any other particle. So, I wouldn't blame you if you think that they are almost impossible to catch. To detect them, if you want a little chance to detect them, you need a really huge detector and a lot of neutrinos. So that sometimes, if you are lucky, 1 out of billions will bump into one of the nuclei of the detector, giving birth to electrically charged particles that can be revealed. So the Opera experiment could be considered the biggest camera ever built in the world. Forget smartphones and think about the old analog cameras with photographic films. There were more than 9 million photographic films in this detector. As ordinary photographic film are sensitive to light, these were sensitive also to all the other charged particles, such as the one produced by neutrinos when they interact. This detector was as big as a building of 4 floors, and it was made of 150,000 bricks for a total weight of 4,000 tons. As I said, it was placed underground in the Gran Sasso laboratories. Yeah, I agree that it doesn't really look as an ordinary camera, but all the bricks of which it was made of were like multi-layer sandwiches of lead, a very dense material, and this special kind of photographic films. So the majority of neutrinos just went through the whole detector. But sometimes some of them interacted with the lead nuclei of the detector and produced charged particles. So these charged particles left a trail in the electronic detectors and told us where to look. So in which brick the interaction took place. And looking at these photographic films, we can reconstruct exactly what happened and understand which type of neutrino arrived to our detector. But why did we need to be inside a mountain to do this? Well, because of all the other heavier non-neutrino particles coming from outer space, cosmic rays. So if it seems difficult to catch a mosquito with a soccer net or even with a mosquito net, as in the picture, it would be impossible if the net was already clogged with bears. Instead, the 1,400 meters of rock above the detector shielded out all these huge beasts and allowed us to focus on our absurd neutrino mosquito hunt. So when a charged particle goes through the photographic film, it ionizes it, and after development, dark dots appear on its path, and we could do a three-dimensional reconstruction of the interaction. With this kind of detector, we can reach a micrometric resolution, which means a resolution of 1/1,000,000th of a meter. To give you an idea of how small a micron is, just think that the diameter of one of your hairs is between 65 and 90 microns. While, for example, a red blood cell is just 8 microns. The width of this picture is about 300 microns. Of course, particles are much, much smaller, but what we see are not particles themselves. We can only see their traces. It's like when looking at the sky, you know that a plane has flown over just because you see the trail. So in this video that I will start in a while, you will see two particles that collide. This is not a neutrino interaction. It's just something to show you how it works to see particles in photographic films. So you will see two particles that collide and produce other particles. So when colliding with an atom, each type of neutrino gives rise to a different charged particle, and we can distinguish them because they behave in a very different way. So we can understand the flavor of the neutrino that arrived at the detector. I patiently analyzed many of these bricks, as done by other 180 physicists from 26 research institutes and universities in Europe and Japan, and at the end of the experiment, we fully reconstructed nearly 7,000 neutrino interactions. And in 2010, we saw the first tau neutrino. We can recognize it thanks to that little red piece of trace that you see near the interaction point, near the vertex. So that little piece of that red piece is the tau neutrino track. So sorry, not the tau neutrino, the tau particle track, that is the one that tells us that tau neutrino gave birth to this interaction. And in 5 years, we found at least 5 neutrinos that were born as muon ones and became tau neutrinos. And in the same year, a Nobel Prize was given to the discovery of neutrino oscillations. Well, unfortunately, not to our experiment. The Nobel Prize was given to the first 2 experiments that saw something about neutrino interaction, the neutrino oscillations. They saw that there were missing neutrinos. There was an amount of muon neutrinos that just disappeared, and they couldn't know what happened to these neutrinos because they weren't able to see tau neutrinos as we could do. So we really demonstrated it, but never mind, the Nobel Prize was given to them. By the end of the experiment, we had found other 5 oscillating neutrinos. The study of these events was part of my PhD, so I'm romantically linked to these ones too, and I have to show them at the end of my talks. So we are at the end, and now you might be wondering if your life will be affected by this discovery about particles that you might not even have known existed before this talk. Well, the answer is no. However, if, like me, you are very curious about the wonders of nature, neutrinos are phenomenal, because these ghostly particles, these ghostly messengers, have been flying around for 14 billion years, ever since the universe popped into existence. So by studying them, we can directly learn about what the universe was like at the moment of Big Bang, and hopefully we can also understand why the universe is made as it is, why it has these properties where you and I exist, and why not other properties where you and I don't even exist. Furthermore, by trying to detect these undetectable particles, we keep inventing new technologies that can take us places where we can't even imagine. Some of these technologies coming from the experiment that I showed you are already used, for example, in medical researches to find advanced cure for cancer, or in the study of volcanoes. So many questions on neutrinos are still open. For example, which are their masses, which is their hierarchy, so which is heavier than the other, if they are the same of their antiparticles, if there exist sterile neutrinos, so other kind of neutrinos. And I hope that some of young people listening us today will help us completing this puzzle. Thank you.
Thank you very much, Juliana, for your interesting and nice overview about neutrino properties, and in particular about the results of the OPERA experiment, which was actually very close to us under the Gran Sasso Mountain. So I do not see at the moment any question in the chat, because people are usually very shy about these topics. But let me just ask you also to mention or to comment, if you want, about the astrophysical neutrinos and the connection of the possible observation of neutrinos with maybe astrophysical sources that are in the last year became very famous because of some connection between the observation of the direction of the neutrinos with some probably astrophysical sources. So would you like to comment on this topic?
Yeah, sure. So today we have this multi-messenger astronomy. It means that we can look at space using different kind of instruments and different kind of particles or properties of radiation and so on. And neutrinos are one of these different kind of ways that we have to watch at the universe. And as I mentioned in the talk, they are unique because they can travel really long distances and they can go through everything. So while, for example, any charged particles can be stopped by a planet or deviated by a magnetic source and so on, neutrinos just go straight. And so they can cross the entire universe. And looking at them, we can understand what happens in the farthest regions of our universe. And today there are some experiments that are really beautiful that look at these neutrino sources and studying the energy and the direction of these neutrinos. They try to understand where they come from, if there are relations with, for example, gravitational waves or some phenomena or some sources that we don't know much today. So I think that within all the eyes that we can have to look at the universe, neutrinos are difficult to catch and to study, but they can give us many answers.
Yeah, so this aspect of the multi-messenger astrophysics is very important and fundamental. So different observation in different fields, like for instance neutrinos and cosmic rays or gamma rays and gravitational waves since a few years. So putting together all this information, we can understand much better, or we can improve our understanding of how these particles and photons are produced in their sources. So then we have to put together the observations with theoretical modeling of the sources, and then with a lot of difficult techniques to really understand what is the direction of these particles, to match the direction of the particles with maybe the direction of or the position of the sources in the sky. So this field is really improving a lot in the last years, and it's really a very trendy topic, I have to say. So I see a question in the chat. Marco is so he's writing a disclaimer about his naive question, and he's writing if there is a consensus about the speed at which I think neutrinos can travel.
Yeah, okay, we measured it and we know that it is almost the speed of light, just a little less, because they have this tiny mass. So maybe the question comes from the fact that some years ago there was a result, a claim from Opera, that neutrinos traveled faster than the speed of light. And this was, let's say, against Einstein relativity theory, because if they have mass, they cannot travel faster than the speed of light. There was a huge mess on this result, but this result was just a preliminary one. The people in the collaboration were still trying to understand if the result was correct or not. But then, you know, some people didn't respect the embargo, so on the newspapers appeared these big titles like "Einstein was wrong, neutrinos travels faster than the speed of light," and so on. So after, I think, just a couple of weeks, we retracted this result because we understood that there was a mistake. it happens. But I think that this is a positive story, because it tells us two things. The first one is that if there is a theory that has been proved to be a solid one, like it is the Einstein theory, if we have an experiment that says something that is opposite to this theory, we don't just throw it in the bin because Einstein said that things can travel faster than the speed of light if they have mass. No, we keep investigating, because if an experiment and then another experiment and then another experiment proves that this can happen, we just need to modify the theory. So there's no like, okay, God said that and we just believe that. No, we keep investigating. On the other side, it also tells us that science corrects itself. So we can be wrong, we are just humans, but in a certain amount of time, sometimes just couples of weeks, we understand if something is wrong, and the big collaboration of people is what makes us sure that we will understand if there is a mistake. So I like, to be honest, this story, even though many people just would like to forget about it.
No, I think it is a very nice example of how, as you said, science corrects itself. And by the way, these violations of the also Einstein's theory are still studied in many fields in physics. So this is not, of course, the end of the story. So physicists investigate a lot also these basic theories and try to put the limits lower and lower to understand, to see what are the limits of these theories that we all accept. So
yeah, just a funny thing, in those couples of weeks in which we didn't know if the experiment was right or not, there were like hundreds of papers written by theoretical physicists that tried to explain this result, to include it in Einstein relativity. So really, theoretical physics is a field where everything can happen. Then we need an experiment to prove if everything is correct or not, as it was for neutrinos. I mean, the idea of inventing these particles was crazy, but this was just an idea until after almost 30 years, it was proved to be right by experiments.
So yeah, so that was a very nice example of this scientific of how science works. So thanks a lot also for reminding us about this story. And so I thank you. I don't see other questions in the chat, so I thank you again a lot on behalf of the TMS committee, and I hope you can stay with us a little bit more. And then I just pass the stage to the next session, that is the chemistry one. Thanks a lot again, Juliana, and
thank you for the invitation.
To see you again, to see you in person at some point. Thanks a lot, and I think
bye to everybody.
Sara.
Welcome everybody to the chemistry session of this Congress. We will start with a plenary lecture delivered by Dr. Stefano Cinti. Thank you, Stefano, for accepting our invitation today.
Thank you.
He is an assistant professor at the Department of Pharmacy, University of Naples, Federico II. He obtained a PhD in chemical science in the group headed by Professor Giuseppe Palleschi at University of Tor Vergata. He leads the Uninano Biosensor Lab, and his research includes the development of electrochemical biosensors, paper-based devices, nanomotors, and nanomaterials. He published more than 45 papers on peer-reviewed journals. In 2018 and 2019, he has been named Best Young Researcher in Bioanalytical Chemistry and Analytical Chemistry, respectively. And in 2020, he has been inserted in World's Top 2% Scientists. He is in the board of the Chemical Cultural Diffusion Group and of the Young Group of Italian Chemical Society. He is active in communicating science to a non-specialized audience through TV shows, radio, and magazine. The title of his lecture today is, Do we need biosensors? Yes, we do. So, when you are ready, you can share your screen and start.
Thank you, Sarah. So can you see my desktop?
Yeah.
Okay. So just we keep this down. So thank you, Sarah. Thank you to the committee for inviting me to deliver this talk. I'm very glad to stay here. So as Sarah said in the beautiful presentation, I am an assistant professor in the University of Naples, and I'm working in the field of analytical chemistry, more in detail in the biosensor. And today I would like to convince you how the world of biosensor can be useful for all of us, for all the humankind as everything. So first of all, I would like to show my path, my academic journey. Then, as Sarah said, I start my PhD, my studies in Rome, Tor Vergata University, and then I had the opportunity to spend a lot of period abroad in Europe and also in the US during my PhD, postdoc, and till now that I got a tenure track here in the Department of Pharmacy. And I just showed this slide to maybe convince younger students that are listening to us today that moving the mobility is the most important things in this work. So if you want to grow, if you want to understand something more, if you want to create something, so you need to go out from your comfort zone, as the expert says. So it was just a suggestion and a little consideration. So let's go into the details. So from an analytical point of view, what we need as humans. So what we want to know? We want to know if we are sick. We want to know if the beverage that we are assuming, the water that we are drinking is clean. We want to know if perhaps some environmental sites are clean, if the sea that where we are swimming is empty of toxic species, and what else. So the point is that when we are going to ask this stuff, so we usually need some experts. We usually need some laboratories. We need personnel. We need instrumentation. So the point is that when you want to obtain an information related to, for example, the presence of some pathogens in food, you need to take a piece of the sample. So sampling your substances, your species of interest, you need to transport, you need to send this molecule, this piece of food in some laboratories. You need to wait for the analysis, and you need to wait also the answer. So I think that the entire process is like more than one day. So and you can apply this kind of thinking to all the analysis that you have in mind. So analysis for health, analysis for environmental, analysis for food, analysis for industry. So in any case, you need to wait. You need to wait, and you need to pay. You need to pay for people that is going to make this kind of measurement. So the point is that a clear advantage is the introduction of the decentralization of the analysis, and biosensor can really be a complementary tool to avoid non-specialized people to spend money, to spend time, no? Because if you think, so if you could have something that you can use every day and in each place you want to monitor something, so could be something better and easier respect to the traditional equipments that are usually used in the laboratories. And the point is that the direction is toward the easy self-testing. So because they are low cost, easy, they don't require a lab, they are portable, and I would like to show you which kind of biosensor exists, which kind of biosensor we develop in our lab here in Naples. But this this necessity is required for important organizations like the World Health Organization that in 2006 established this kind of guideline for each of the diagnostic tests. And this acronym is very important because it tells us how our portable diagnostics should be affordable, sensitive, should be user-friendly, should be equipment-free. And in any case, this is going to lead to integrated, rapid, sustainable response that are in opposition with the traditional methods that usually are expensive and sometimes produce a lot of waste. But what is a biosensor? Oh, okay. Here I just put an image on an old school biosensor. So I think that most of you know about the story of the canaries, of the birds using the coal mine by the miners to understand, to sense the presence of gases, toxic gases like carbon monoxide. So the point is that the little birds was one of the first example of portable biosensor, just because if the gases was too much, so and we cannot sense its presence, so the bird was able to sense the presence, and then if the bird died, so the point is that the miners had the time to go out from the mine. But of course, this is not the best choice. This is not the best choice for several reasons. One reason is the ethics. One reason is the point that a lot of species are heterogeneous, so the response could not be the same. So the sensitivity could not be the same. We need something more, and we have something more, something better, something easier. And here I just posted some of the most famous examples: the blue glucose test, the pregnancy test, and now is very actual, the COVID test. So these are the most important and the most famous examples about biosensors. And in very practical way, you can monitor whatever you want. And these are some examples. These are to be honest, the example that had more fortune to reach the market for several reasons, even here. But what is a biosensor? How is made? So biosensor is made essentially with three parts. So there is a biorecognition element, there is a transducer, there is a signal. So when we think through biorecognition elements, I would like to highlight that, of course, if we are talking about biosensor, we need a bio element that is capable to recognize a particular target in a specific way. As you can see here, this biorecognition element can recognize the triangle, but is not able to recognize the circle. Of course, we can also develop sensors that has at the same time a recognition element that is not bio. Okay, so you can imagine polymer nanoparticle that I will show you later a slide. And also you need a transducer. You need to understand, you need to visualize the biorecognition, and you can do this in several ways, from exploiting a colorimetric change on a redox reaction, vibrational sensor, or fluorescence, or what else. and you need to read this signal on a display, for example. But in the detail, what we develop in the University of Naples here in the Uninano Biosensor group is the electrochemical biosensor. Why electrochemistry? So the point is that what we want to do is to observe a target after a redox reaction occurs. So in this case, so we have we can choose a lot of recognition elements, recognition or biorecognition elements. So the importance is that after the recognition of the target, we have the generation of current or the change of potential. This this follows a redox reaction. And what is the essential part here in this method? In this method is this orange point that is an electrodes. An electrodes is just the last part of the sensor, and is also that component that is capable to be modified depending on the necessity, because electrodes is where the redox reaction happens. And here we develop a special kind of electrochemical biosensors that are made on paper. Here I just reported the procedure to realize. I'm not going into the detail, but I just would like to spend some word on the use of paper, because the traditional electrochemical biosensor, portable electrochemical biosensor, are realized on plastic, on glass, on alumina. So the point is that paper is very in line with the concept of sustainability that we are required from the organization, from the Europe Union and what else, because also the possibility to use paper as the substrate, we're realizing this kind of electrodes that are screen printing electrodes that are obtained with serigraphic printing. That is the same technique that you use, or you have seen printing some t-shirt, for example. So in this case, we use some kind of inks that are conductive. Perhaps they can contain graphite, they can contain silver, they can contain gold. So the important is that these inks are conductive. And here, this is the final results. So we have this kind of strips. So don't be worried about the real meaning of this. So what you should understand is that here in this area you can build your sensor. This is just this is just a versatile platform that you can build like building block, choosing which is the biorecognition or the recognition element that you need to detect some molecule. This is, for example, so here is the versatile platform, the screen printed electrodes, and you can modify this with various materials. So if you want to realize a biosensor, you will need some enzymes, some antibodies, some nucleic acids, some cells, whatever. If you want to develop sensors, so excluding the bioelements, you can use ionic liquids, nanoparticles, metal nanoparticles, or carbon-based nanomaterials, graphene, carbon nanotubes, what else? As Professor Savage, so you can also build here a nanomachine, for example. So you have a lot of possibility also to bring together these two worlds of non-bio and bio. So you can make a sensor with nanoparticles and enzyme, because maybe nanoparticles gives you some feature, enzyme gives you other features, so you can mix depending on what you want. And why paper-based diagnostic? Because paper is abundant, is sustainable, is versatile, is multitasking. You can build only one platform in paper. What do I mean when I say only one platform? So paper has a great advantage, that is porous, and this porosity allows to you to do several stuff within its structure. So you can load all the reagents for an assay in the paper. You can take paper and maybe develop two-dimensional, three-dimensional origami-like structure platform. You can use paper to filter. No, the chromatography is based. The paper chromatography is based on this, on the capacity of paper to retain some molecules that are bigger than the pore size. And you can use also paper as a reactor, so to make some reaction happen. So the word around paper as a new kind of support for this kind of electrochemical biosensor offers up tremendous possibilities. And what I would like to do now is to show you some example that can make you more confident with this word. And for example, here we realized a sensor for the phosphate ions in a river water, which was here the advantage. So here is the lateral section of the paper, what I highlighting here, and the red stars means that it is a kind of molecule that you need to carry out a assay. So in this case, the red stars is the molybdenum salt, acidic ions, supporting electrolyte for the electrochemistry transduction. What happened is that in this case the phosphate is recognized by the formation of a phosphate molybdenum phosphate molybdate complex that is indicated by the yellow star. And this can happen when the phosphate that is contained in the sample that you want to analyze, for example, the river water, is going to wet the paper, and then is going to create a chamber of reaction. And as you can see, the phosphate that is the green circle is going to react with the red star, creating the yellow star that is the electroactive molecule. So it's the molecule that you can detect following a redox reaction. And as you can see here, we developed this with different concentration of phosphate in the river water, and this was one of the first example that we carried out with paper. Then we move ahead. So we applied this kind of strategy to ethanol and beer, and in this case, as you can see, there is a mechanism that involves the use of biosensor. So here there is an enzyme that is the ethanol, this the alcohol oxidase. Here there is a nanoparticles made of Prussian blue. Why? These two have a synergistic action, because in presence of ethanol and oxygen that is naturally dissolved in the beer, so the enzyme is going to oxidize the ethanol, and one of the byproducts is the hydrogen peroxide. Now hydrogen peroxide can be reduced in presence of this electrocatalyst that is the Prussian blue, and this is going to have like a catalytic cycle. Okay, this reduction produce a current at the electrodes. So imagine that all these enzyme, all these nanoparticles are immobilized here. So at the end you have just one strip that contains all what you need to carry out the analysis. And here we build just a calibration curve, and you can see that these are the current that your output, the current deriving from the oxidation of ethanol, the production of hydrogen peroxide, and the consecutive reduction of the hydrogen peroxide at this nanomodified electrode. So more is the ethanol that is present, more is the hydrogen peroxide that is produced, more is the cathodic current that is produced. The cathodic current is related to the reduction of this. So more the curves go down, more you have a signal, and you can quantify in a long range different beer, and you can classify, especially for the quality control, in the industry, but also for the consumer that wants to know if a beer is, for example, without ethanol, or there is too much ethanol, so a lot of application. Then we apply a similar concept to the chloride detection in sweat and serum by explaining this kind of reaction in presence of chloride and silver. We can just form the silver chloride, and this is made through a redox reaction. So what we can do is just take a drop of sweat, drop of serum that normally contain chloride, and then following this kind of reaction we can see this kind of signal. So don't look at much of this signal, but what is important is that here this is a cyclic voltammetry. What we can see from left to the right is the presence of a peak, and this peak is like an oxidation. So when silver and chloride are present, so you have an oxidation, you have the formation of silver chloride, and going back you have the reverse reaction, so a reduction, so the ions are formed back, and we apply this in serum and sweat, and this can be a very very good application, for example, for cystic fibrosis. So one of the gold standard method for detecting, for diagnosing, and for monitoring the care of cystic fibrosis is the monitoring of chloride ions in sweat that are very, very, very high in this kind of patients. And this can be a really affordable, really user-friendly tools. And here you can see in this image the presence of the sensor on paper. Then recently we just published the detection of mercury in drinking water. In this kind we use a sensitive sensitive element, gold nanoparticles, because gold nanoparticles are able to form an alloy with mercury. So with stripping methodologies, that is something a little bit more complicated than a voltammetry, but here I just reported the mechanism. So first of all, we apply a difference, potential difference, because we want the mercury is absorbed, is reduced onto electrode, and after we are going to redissolve, we are going to strip the mercury, so putting it in the solution, then we can observe a signal that is proportional to the presence of mercury. In this case, we used two kinds of paper, so an office paper where to print the electrons, and a filter paper combined in a 2D, in a three-dimensional architecture, because the filter paper can pre-concentrate the mercury ions, and here is a calibration curve in presence of a classic sensor office paper chip in presence of this different concentration of mercury. And here I just show that in this small inset how the 5-per pre-concentration with the filter paper can enhance the sensitivity. Here you can see that the red line is without pre-concentration, and it will be this line, and the black line is after 5 steps of pre-concentration. So paper without any cost allows you to pre-concentrate sample, and from an analytical point of view means that you are going to enhance the sensitivity, so you can detect lower and lower concentration. And we are very happy because this work has been highlighted in the cover page of this journal, so it's a good achievement for us. Then I always other example, organophosphate pesticides in soil, and here should be written food extracts. But here we develop a enzymatic biosensor based on an enzyme, the butyrylcholinesterase, that is inhibited by the presence of organophosphorus pesticide. So what we are going to do now is that we take our electrode, we modify this with enzyme, with the mediator, with the electrocatalyst that we need, and we are going to follow the enzymatic activity in absence of the inhibitor, that in this case is the pesticides. So we are able to monitor the production of the thiocholine, that is a thiol that we can recognize at our electrode. And what happened is that if the inhibitor is present, the enzyme is not going to work enough, so you have a lower production of the substrate, so you have a decrease of the signal, so you can correlate the presence of the inhibitor to the inhibition of the enzyme. And this can be very useful for inside application. Then I will just to conclude with two of example for other example in clinical sets, because it's very important. This is a new line of research in our lab, especially the development of sensors and biosensor with the use of synthetic probes like oligonucleotides, peptide nucleic acids, and what else, to detect circular tumor DNA associated with breast cancer. For example, here we developed two different strategies called signal on or signal off that are different because in the signal on you have a signal that increases when the target is present. In the signal off you have a signal that decreases when target is present instead. And I'm not going to the detail because it's a little bit long, but if you want you can read the paper, you can ask me, you can send email, and I will be happy to talk with you about this. But what the principle is that we are going to take our electrode and we are going to immobilize the probe. This probe can be with an electrochemical mediator inside or without. And when the complementary DNA is present, this is going to hybridize with the probe. And then we are able to understand and to visualize this kind of recognition, this complementary recognition. And we apply this in buffer, in blood, and we compare just these two kind of strategies. Last example is liquid biopsy of microRNA for pancreatic cancer. In this way, we used here to modify the electrodes with gold nanoparticles for enhancing the signal, with PNA, because PNA is without charge, it's not charged. So the method was based on the fact that PNA is able to recognize the microRNA, the complementary microRNA that we are searching. And in presence of microRNA, the concentration of negative charge on the electrode is higher than in absence. And the point is that we can recognize this applying this by adding an external electrochemical mediator that is absorbed on electrostatic interaction. Here also we test a different range of this concentration of microRNA in a serum and buffer sample, and we applied also to real sample. So I would like to ask with the question that it was part of my title. So I asked you, so do we need biosensors? So I think that in any case, we need we need biosensor, we need portable instruments, because we need to take citizens and make them more responsible and more active to the society, the famous concept of the citizen science. So I think that if each of us can have some instruments easy to use to monitor how a river is clean, how a food is good or is not is in the shelf life period, or if you can monitor your cholesterol, for example, daily, so you can save a lot of money, a lot of time, and you can give a real support to the society. I would like to thank all the Uninano Biosensor group that I lead. Here there are all the students that are helping me to produce this kind of results. So, and they are very passionate in this kind of activities. And also I would like to thank my department, that is a department of excellence, that allowed to me to get this position in Naples. Then also the Marie Curie for some funding, and also Fondazione Umberto Veronesi, especially for the breast cancer activity. So thank you very much. I would like to share my okay. I just finished, so if you have question, I'm available.
Sorry, I didn't have the audio. Okay, so there are some questions. For first, thank you for your presentation, very interesting presentation. And I think that biosensors are a very actual topic. So we have a question from Marco Ponsetti. Could you use this technology to produce a reusable ethylometer that you can fit in your wallet in place of a credit card?
Okay, thank you, Marco, for the question. So the point is that in theory we can generate even smaller than to be able to be inserted in a wallet. The point is that in this case, for an ethylometer, so we should consider that what we want to, where we want to sample the ethanol. So the breath, for example, so or a liquid, or for example sweat. So for this, in another application that we're working on is the production of a wearable sensor for the ethanol in sweat, to be more sensitive respect to the current ethylometer, and to be non-invasive. But yes, the answer was yes, we can produce this.
Okay, thank you. Next question from Luisa Giansanti. Which are the advantages of electrochemical sensors with respect to colorimetric or other sensors?
Thank you, Luisa, for the nice question. So there is a great there is a main advantage in using electrochemical sensors. So the point is that electrochemical sensors are blind to the matrices. So for example, if you're going to analyze blood, serum, or some colored matrices, we are not going to be affected by the presence of color, while the colorimetric test can be affected by this. So you should, in this case, you should treat the sample, dilute, and what else. So this is the main advantages, that you need. You don't care about the color of the solution. This is very important in when working with real matrices. Of course, I want to be honest, because also the colorimetric tests are very useful and they are very good features. For example, colorimetric test, and we also develop colorimetric test here in our lab, have the advantages that most of the time don't have to require a reader, for example, so you can just naked eye visualize a results. Instead, the electrochemical one need a reader that now can be also associated with a smartphone. So I think that there is not the best platform. I think that depending on what you need, if you're going to work with a very complex, very colored solution like blood, I would suggest to develop your electrochemical sensor. If you're going to work with a clean solution, if you can develop a colorimetric test like a pregnancy test, like this kind of visualization with the smartphone, so I think that I would suggest to work with this.
Okay, another. Are there any side effects of using the biosensor, since it can react with the human biological system?
Okay, thank you, John. Thank you for the question. No, there is not a direct interaction. Okay, so for example, if you had in mind the glucometer strip for diabetes patients, so if you think you can just take a drop of sample and put on the strip, so this kind of biosensors are not going to go into our body. Okay, maybe this is the next step for the implantable one, but this kind of point of care have not a direct interaction. So we need a drop of sweat, a saliva, urine, we need tears, we need this kind of stuff. So there is not a side effect. Of course, when we are talking about wearable, for example, we should consider if on the strip there are some components that can be toxic for the human body. So of course this should be evaluated case by case.
Okay, another question from Marco. He thanks you for your interesting and engaging talk, and he says that seems to understand.
It's actually from Armando Carlone. It's from YouTube. I'm just copying and pasting.
Okay, from Armando Carlone. Okay, sorry. He seems to understand that a limit of your innovative and creative solution for biosensors, as for other probability, is the use of aqueous solution. It would be great to apply it to organic media, as to detect compounds of interest in the downstream media of industrial process.
Oh, thank you, Armando. Thank you, Armando. Yes, this is a great implication in using this kind of paper-based sensor, even because the conductive inks that we use are made with organic solvent. So the point is that if you want to use this technology to highly organic content solution, we risk to dissolve the inks. So we are moving toward the possibility to apply this kind of paper-based, maybe using water-based ink to make the electrodes, and to be applied, as you suggested, in the industrial setting. So to monitor so solutions that are not water-based, but also other possibility are just to use other kind of material, different of paper, even flexible, even cheap, that maybe can resist more, that can be more stressed in this case of in presence of this kind of solution.
Okay, thank you. Now I have one curiosity. Are there any disadvantages related to the use of biosensors or related to the signal detection techniques?
Okay, thank you, Sara. I think that the main limiting point of this technology is represented by the transduction and by the miniaturization of the systems. But I want to say that there are a lot of companies that are producing year by year new device, even smaller. So, for example, there are some companies that are producing some reader that are compatible with almost all the smartphone that can be used by everyone. So I think that the main drawback is sometimes related more to the analytical point of view, because of course, as I said before, the complex, the advantage with respect to the colorimetric ones is that we do not suffer about color of matrices, but of course in the solution there are also other kind of interference. That, for example, if you are going to analyze some oxidation in blood, so you should be aware that there are some antioxidant and something else that can interfere, even if they are not colored. So I think that the real challenge is to find the real specific probe, the real specific element, and also avoiding the use of biological component, instead using molecular imprinting polymer, aptamer, or something like this, because biomimetic peptides, because one of the first limitation is the duration, is the shelf life of this kind of bioelement when they are in the dry paper. So these are more or less the limitation.
Okay, perfect. So I think there aren't more questions. So thank you another time for your participation, Stefano.
Thank you.
So let's move on with the selected oral contributions. The first is by Francesca Di Donato from University of L'Aquila. The title is HSSPME, GCMS, volatile fraction determination, and chemometrics for the discrimination of typical Italian pecorino cheeses. If you are ready, Francesca, you can share your screen and start.
Can you see my screen? Can you see my screen?
Yes.
Okay. Good morning, everybody. I am going to show you an application study using the headspace solid phase microextraction coupled with gas chromatography mass spectrometry for the discrimination of typical Italian pecorino cheeses. Most varieties of pecorino cheeses produced in Italy have a strong geographical identity because the typical cheese-making conditions have a, can reflect the traditional know-how.
Francesca, sorry, maybe you can talk closer to the microphone because I think that we cannot hear very well.
I think we're having some technical issues.
Yes, probably yes. I try to ask her.
Okay, maybe while Francesca is trying to fix the issues, we can go to the next speaker, and then we can come back to Francesca.
Okay. So next presenter is Claudia Marconi from University of Roma La Sapienza. The title of the presentation is The Response of Lysozyme to Oxidation by Hypochlorite. Claudia, are you here? Okay. Okay, we can see your screen, so you can start whenever you want.
Oh, sorry, I deactivated you by mistake. Okay.
Now, can you hear me?
Yeah.
Yeah?
Yes.
Yes, okay. Good morning. I am. Can I go?
Yes, please go ahead.
I am Claudia Marconi. I am a PhD student at Sapienza University of Rome, and I will present this work that I studied in my degree thesis. And it is the response of lysozyme to oxidation by hypochlorite. Lysozyme is a protein that has an antibacterial function, and it is part of the immune system response to an external pathogen attack. But also, this is the secondary structure of this small molecule, but also hypochlorite is part of the immune system. In fact, also hypochlorite is produced by activating the leukocytes. But in some cases, such as in chronic disease, chronic infections, there may be an excess of hypochlorite, of production of hypochlorite, and this potent, powerful oxidant can oxidize protein themselves, host protein themselves. So protein that undergo oxidative stress can lose their biological functions, but also misfold, and this can lead to the formation of insoluble deposits, such as amyloid fibers. So the idea to study this process is that proteins has oxidizable components that can be oxidized selectively because they are a different kinetic constant. So we can oxidize selectively by controlling the molar ratio between oxidant and protein. So I added hypochlorite solution to protein solution, and I observed the formation of a precipitate, as we can see in these photos, for the higher molar ratio, oxidant protein molar ratio. In order to study this phenomena, I make some experimental measurements. Here is UV spectra, and we can see that immediately after the reaction, there are no such great difference, but with time, there is an increase of the difference of the modification of the spectra. These are the oxidized solution, but also we can see the same thing in the filtered solutions. So in the solution that I passed through a desalting column, so I eliminated all the hypochlorite and other products. There is a modification in the time, after hours. Then I did circular decreasing measurements, and also with this technique, I observed a modification in the curves with the higher molar ratio. So I analyzed this spectra, and I find out that there is an increase in the beta sheet content in the protein and a decrease of alpha helices. So a modification in the secondary structure of the protein with the molar ratio greater than 4, even in this case. With dynamic light scattering, I observed an increasing of the derived count rate that is proportional to the formation of the aggregates. But in the filtered solution, immediately after the reaction, I saw only the monomeric signal of lysozyme. But also in this case, with time, I observed the formation of larger aggregates with the higher molar ratio, oxidant lysozyme molar ratio. So I did, to study the nature of this precipitate, I did the thioflavin T assay that is considered an assay specific for amyloid fibrils. And in this case, I had a positive response, both in the oxidized and in the filtered solutions. So I studied the precipitate also with the X-ray diffraction, and also in this case I observed two peaks that resembles in such a way a literature spectrum of amyloid fibers. So there are these two peaks that can be related with the amyloid fibers. And then I make also microscopy, atomic force microscopy, and this is one of my samples with the higher molar ratio, oxidant protein molar ratio, and we can see some little aggregates, but also some strange structure at the edge of these aggregates. And then I compared it with an amyloid fibril sample that I did myself, and we can see that these structures are very similar. At the end, I would like to emphasize the importance of the mixing method, In fact, here is an experiment where I prepared the same sample, that is the same final molar rate, the final concentration, the same, but in a different mixing method. And we can see that if we add a dilute hypochlorite solution to a concentrated lysozyme solution, we obtain the less content of aggregates. So we can state that there are no local gradient concentration that can oxidize the various molecules, not in the same way, not in a uniform way. So I can conclude stating that the oxidation by hypochlorite triggers a phenomenon of supramolecular aggregation, and there is a critical threshold that was in my experiment around 4 with the oxidant lysozyme molar ratio. And but I observed that there is a great influence of the mixing method, just as we have seen. So the future perspective are that we can adopt an optimal mixing method, so it will be possible to investigate the structural and conformational variations of the protein and the nature of the precipitate in a better way. So I can stop here, and thank you all of you for your kind attention.
Thank you, Claudia, for your very clear presentation.
Thank you.
So, are there any questions? Don't be shy. Okay. A question from Luisa Giansanti. Can you use other proteins, for example, albumin?
Yes, we can use other proteins. Albumin has been used and studied in another works in my group, in my research group, but the studies is now going on, so we can wait for the results. But yes, we can study other types of molecules, proteins, etc.
Okay. Any more questions? Okay, another question from Giovanni De Gasperis. Which software are you using to visualize AFM surfaces?
There is a software with the instrument. I went to the CNIS, the Center of Nanotechnology for Engineering of Sapienza, to make this IFM microscopy measurements. And the software was with the instruments, and there was people that used it. So I was only a spectator. She gave me the results, the photos and the results.
Okay.
The inputs, and she did the photographs.
Okay, so if there aren't more questions, we will try to connect another time with Francesca Di Donato. Francesca, are you here?
Yeah.
Okay, so you can try to share your screen.
Yeah. Okay.
Okay.
Okay, do you see my screen?
Yes. Perfect.
Okay. Good morning again, everybody. I'm going to show you an application study using gas phase, solid phase microextraction coupled with gas chromatography mass spectrometry for the authentication and discrimination of 3 typical Italian pecorino cheeses. Most varieties of pecorino produced in Italy have a strong geographical identity because the typical cheese-making conditions reflect the traditional know-how of the production area. This relationship between the cheese variety and the production area has been officially recognized by the attribution of the European and international certification, like, for example, protected designation of origin as low food presidia. It is the case of Pecorino di Farindola, Pecorino Romano, and Pecorino Sardo. These certified products present higher commercial value, therefore developing analytical methods for their authentication and tracing is a necessary tool to protect their label. In general, pecorino is an Italian dairy product obtained with used whole milk. Milk can be coagulated with different types of rennets, and after its formation, the curd can be cut and cooked. After whey drainage and salting, then the cheese is ripened for a typical amount of time, in which its chemical composition and organoleptic properties, flavoring ones above all, can evolve. Looking at this process, we have identified the coagulation step as the point of the greatest difference. According to the cheese-making protocols, indeed, pecorino di farindola is made by means of pig rennet, whereas more stable rennets, such as lamb, calf, and calf past, are used in the production of pecorino romano and pecorino sardo. This difference could lead potentially in variation in the volatile organic compounds profile, because these compounds are generated during the ripening time, mostly by lipolysis that concern the liberation of free fatty acids, and proteolysis concerning the casein matrix degradation to give peptides and free amino acids. The most suitable technique to analyze the volatile organic compounds is the head-space solid-phase microextraction. During the extraction, the compounds generated by the sample saturate its headspace and interact with the fiber in a dynamic equilibrium of absorption and desorption. After sampling, final desorption took place in the GC injection port with a mass spectrometer as detector. Chemometrics was applied in this work to achieve different objectives. Upstream the experimental investigation, we used a design of experiment combined with response surface methodology to plan to define the experiments to be performed in the experimental domain and optimize the analytical protocol. Downstream, we applied a multivariate classification approach to answer to the question, is this product compliant with this label? So the final purpose of this study was to build an analytical and chemometric protocol for the discrimination of these three certified pecorino cheeses produced in central Italy. To achieve this objective, we analyzed 47 samples of pecorino cheeses by means of headspace solid-phase microextraction coupled with gas chromatography mass spectrometry, and finally we classified them with a discriminant approach. First of all, we applied a 3-level full factorial design to optimize the sample temperature and exposure time of the fiber to the sample headspace. That are the two variables that can potentially affect the extraction. Two responses were calculated and maximized with the mathematical approach of desirability function. The response surface generated by the regression of these D values can show the point in which this D values is maximized. The compounds previously extracted were analyzed by means of GCMS, and we have identified 39 compounds that can be represented by the families of acids, carbonyl compounds in which ketones largely prevail on aldehydes, esters, alcohols, and other minor components such as terpenes, ethers, and olefins. The same information can be easier visualized with a projection of the sample in the first and second principal components space, where you can see a clear separation of the PF samples and the less evident ones of the two classes of PS and PR. This less evident differentiation can reflect the common origin of the milk, that is exclusively for PS and predominantly for PR from Sardinia region. The sample behavior can be interpreted looking at the distribution of the variables on the right. And for example, we can say that the clear distinction of the PF sample can be ascribed to the alcohols that indeed largely prevail in PF cheeses and that lie where the class is localized. Eventually, we have classified these samples using the partial least square algorithm combined with discriminant analysis approach. The PLSDA model was calibrated on a training set of 31 samples and then applied on a validation set of external samples, collecting the 16 remaining ones. Looking at the graphical result of the PLSDA analysis, we can say that there is a clear distinction of all the samples belonging to the three classes, with exception of the two misclassified samples that are one of PF class and one of PR class. Then looking at the variable importance in projection, the VIP, we can also evaluate the variables that contributing the most to the model, that are the first 14 compounds in the list. Concluding, we can say that the analytical and chemometric protocol built in this work was found to be suitable for the discrimination of these three certified products of the Central Italy. And we can say that also that only two of over 47 samples have been misclassified. I would like to thank the organizer for allowing me to present my research today and all of you for your kind attention.
Okay, Francesca, thank you for your very nice presentation. Are there any questions or curiosities? Okay, waiting for questions. I have one curiosity. Did you investigate more about this kind of cheeses, or I mean, did you perform other different analysis or studied other characteristics?
Yes, we did. We have also investigated the mineral content and the fatty acids profile to have a complete picture of these pecorino cheeses. And in the meantime, we have also analyzed this sample with mid-infrared spectroscopy, because this is a technique simpler and faster. So the best result would be obtain the same results of the other techniques, but with this simpler technique.
Okay, sorry. Okay, another question from Michela Ciocca. Can you apply this analysis also for other products?
Yeah, sure. In my lab, we are studying many traditional foods products, like for example, saffron, wine, other foods. Yes, it's possible. Also, not food to food, but to other stuff.
Okay, thank you, Francesca. If there are any more questions, I pass the floor to Marco for continuing.
Okay.
Okay, thank you very much. So now we will have a short presentation from the sponsor of the event, which is Aurogene. Before that, I would just like to show you this slide, if I manage to share the screen. There you go. Okay, we have a votation going on for the best elevator pitch. So you can follow the slido.com link and insert this code to vote, or the link I pasted in the chat, or alternatively, you can scan the QR code to vote. Okay? So I'll just give you 10 seconds to scan the code. Okay, so now I will give the virtual floor to Sofia from Aurogene. So let's see if she manages to activate. Okay, there she is.
Buongiorno a tutti.
We will also change language for a moment because Aurogene is an Italy-oriented company, so we will be speaking Italian for a few minutes. Thank you.
Schermo in quanto vorrei spendere qualche parola sull'azienda che è Aurogene. Allora, posso chiedere la conferma se vedete lo schermo?
Sì, sta arrivando, la connessione è un po' lenta, ma sta arrivando.
Scusa che sono in location un po'... Mi sentite?
Sì.
Ok. Ok, allora vi ringrazio di nuovo di averci invitati a partecipare a questo evento e oggi in merito alla giornata che era presentata da TMS vorremmo parlare di Aurogene, che innanzitutto vorremmo farvi conoscere chi siamo, da dove veniamo, quali sono la nostra mission, appunto il nostro scopo e di che prodotti ci occupiamo sul territorio nazionale. E come potete vedere in questa slide, Aurogene è nata 15 anni fa con base a Roma ed è stata fondata da ex ricercatori, quindi i miei responsabili hanno concluso gli studi di dottorato e si sono avventurati in quella che è la distribuzione dei dispositivi per la ricerca scientifica sul territorio nazionale. A giorni d'oggi siamo presenti in 12 regioni in tutta Italia e riforniamo non solo le università italiane ma anche i laboratori privati. Infatti l'ultima novità è che ci stiamo espandendo anche nell'ambito della diagnostica. Tra le università che sono i nostri clienti abbiamo l'Università di Sapienza di Roma, la Bicocca di Milano, Università di Milano, ma anche gli enti di ricerca come Humanitas, di ricerca a Genova, San Martino, l'ospedale San Martino e collaboriamo con davvero molti brand, ce ne sono più di 30. Il nostro maggiore focus di prodotti è sulla biologia molecolare e biologia della cellula. Immagino che tra di voi oggi ci sono anche dei ricercatori che si occupano di questo ambito di ricerca. Allora, la nostra missione è quella di appunto semplificare e rendere più facile la vita di tutti i giorni dei ricercatori e applichiamo quello che è l'approccio individuale e cerchiamo di trovare delle soluzioni che sono più adatte a miglior prezzo, quindi che sono vantaggiose dal punto di vista economico. Come ci impegniamo ogni giorno per servire alla scienza? Diciamo che partiamo da quello che è il supporto alla biologia molecolare, quindi estrazione ed amplificazione degli acidi nucleici, estrazione e rilevazione delle proteine, analisi in Western blot, poi analisi anche dell'espressione delle genica, quindi PCR real time. Inoltre distribuiamo i prodotti come anticorpi, i kit ELISA e dispositivi per analisi in epigenetica. Qui riassumo le ditte di maggiore interesse, soprattutto quelle di cui siamo distributori esclusivi, come Bioline, di cui abbiamo i prodotti che riguardano appunto lo studio degli acidi nucleici sia DNA che RNA, e il nostro recente partner che è Mirxes, un'azienda di Singapore che si occupa di studio dei microRNA, quando sappiamo che negli ultimi anni lo studio della biopsia liquida è uno dei cavalli da battaglia dei ricercatori. Tra le altre nomi potete trovare la Zymo Research, Norgen, ABM Good, ci sono dei prodotti con marchio anti-aurogene e Advanced che sono principalmente concentrati sul studio delle proteine in Western blot. Qui invece potete trovare le ditte sempre principali, non sono assolutamente tutte, per quanto riguarda gli anticorpi, quindi la Santa Cruz, BioLegend che si focalizza sui anticorpi per la citofluorimetria soprattutto, e poi abbiamo ditte che rappresentano i kit ELISA come Maptec, Betil, Cusabio, Southern Biotech sempre per gli anticorpi, e inoltre Invivogen che ha un vasto repertorio di prodotti per la biologia cellulare, soprattutto decontaminazione come da micoplasma, che sappiamo ben essere un grave problema a livello dei laboratori di biologia cellulare. Inoltre ci occupiamo anche della parte di plasticheria. Oltre ai reagenti, ci occupiamo di fornire dei servizi che sono il sequenziamento in NGS, sintesi dei peptidi e anche degli anticorpi e la profilazione della metilazione del DNA. Per quanto riguarda il sequenziamento in NGS, collaboriamo con la ditta coreana DNA Link e servizi di epigenetica via Keynote. Non ci fermiamo al reagentario perché abbiamo anche la possibilità di rifornire i laboratori con la strumentazione che sia piccola o grande, quindi partiamo da centrifughe, spinner, termosmiscelatori, lettori di piastre, ad esempio per analisi ELISA, i termociclatori, ma anche i macchinari per la real-time PCR. Infatti durante la pandemia, il Covid-19, siamo stati molto presenti sul territorio nazionale per quanto riguarda i macchinari per l'analisi appunto del SARS-CoV-2. Poi per completare il quadro ci occupiamo anche di grande strumentazione che sono freezer, frigoriferi, cappe e incubatori. Vedete appunto i principali marchi con cui collaboriamo in questa slide, quindi Ermle, Azu Biosystem e BioAir. E con questo credo di aver concluso, e se ci fossero delle domande, curiosità, resto a disposizione. Io non mi sono presentata all'inizio, non sono specialista di vendita a livello di aurogene e siamo lieti di accogliere le vostre problematiche e trovare la migliore soluzione ad esse. E ringrazio tutti per l'attenzione.
Ok, grazie mille Sofia. Adesso possiamo passare, se vuoi puoi scrivere in chat il link Auto-Gene.
Sì, mando il link del sito.
Perfetto.
Ok.
Now we can switch back to English. Thanks again to Origin for sponsoring the event. And we now go to the computer science session. So I'm going to activate. There we go. Okay. Thank you. Go ahead.
Good morning. Good morning. Good morning, everyone. My name is Ivan Letteri. I'm a postdoctoral fellow at the University of L'Aquila. Today, I'm here to present you Dr. Giovanni De Gasperis, Assistant Professor of Cognitive Robotics since 2007 at the University of L'Aquila in the Department of Information Engineering, Computer Science, and Mathematics. Now, quickly a few things about the curriculum of Giovanni. Curriculum that proves his skills about the topic of what he is going to present. Giovanni graduated in electronic engineering in one thesis on application of neural networks in automatic control. Then from 95 to 98, he was a postdoctoral fellow at the Anderson Cancer Center of the University of Texas. Since then, he has been teaching many introductory and advanced courses in computer engineering and science, including test mining. In 2020, he taught text mining with Python in the second level master in big data, statistical methods for knowledge society at the University of Rome, Rome La Sapienza. Giovanni is a member of a lot of associations related to artificial intelligence, like National Laboratory in Intelligent System and Artificial Intelligence, and member of Italian Association for Artificial Intelligence and many other associations. To conclude the presentation, I like to mention that Giovanni spent various periods of study and research abroad: University of Texas, Houston, University of Westminster, London, UK, University of Salamanca, Georgia, and many other important institutions. Now, and before handing to Giovanni, I would like to invite you, attendee, to write your question and comments in the chat to your right side. At the end of the talk, there will be time for question and discussion. So thank you. And now, Giovanni, the air is yours. Please go ahead.
Thank you, Ivan. Very interesting presentation. I didn't know about that, all of that things. But anyway, yes, I'm trying to share my screen with this browser. Okay, so we'll start the presentation. And I'm going to present the Python ecosystem for science. I don't know why it doesn't start. Maybe because I have two monitors. I just switched off one. So you will guess which one is the right monitor. Okay. Maybe I should restart the share screen. Okay. It's very... Okay, interrupt. I'm sorry about this, but as usual, technology takes over. I'm usually in control, but this is the demo effect. Ok, now let me restart this. Ma sono ancora online? Questo mi ha cacciato fuori. Cioè, mi ha cacciato...
Ti vediamo.
Mi intervieni e di che sono...
Seems there is some problem with the microphone of Giovanni. Maybe we can try to wait simply waiting some second. Send some message if you solve it, if you fix it. Giovanni. We simply need to wait some seconds. I received a message from him, from Giovanni. He said that he's starting to log in again.
Maybe if that doesn't work, he can send you the slides and you can present with him talking. You can sure talk over.
Or there is another solution. I work with Giovanni. Okay, there is. So I will close audio and interrupt the video.
Okay. You should see my presentation now. I hope so.
Yes.
Sorry.
It's almost there.
Maybe I should start with another. Next time I used to use a Linux computer. Huh? Windows is always leaving me behind. Okay, let's go forward. Algorithm. Before starting talking about computer science and any programming language, I have to clarify a few fundamentals, because otherwise I'm not sure if everybody is aware of what a computer can do. So you can actually look it up, the algorithm definition from Wikipedia. The most important thing is that an algorithm is a finite sequence of well-defined computer implementable instructions. So it's not a recipe like you cook your spaghetti, but it's something very non-ambiguous and very precise that the computer has to follow step by step. And usually it's done by a CPU which only understands the machine language, which is only a binary code, okay? And also don't confuse an algorithm with an heuristic, which is more like a method that we use in our laboratories for practical things, and we use estimates and Gaussian distribution of numbers. A computer needs to have exact values, okay? That's important because we are now going to see what is a high-level computer language, computer programming language. From my point of view, this may not be the perfect definition in terms of theoretical aspects, but definitely is what I would like to, the perfect computer programming language, high-level computer programming language should be. First of all, it's textual, okay? So far, nobody has been able to program a computer using a non-textual language. So maybe in the future, maybe. It should be readable, definitely, first of all, by a machine, right? I already told you. It should be non-ambiguous and it's context-free. You may look it up, the theory of languages from the scholars of linguistics, and you know that context-free, it means that each word has its own meaning and there is no other words in relation to it that can add meaning to it. So this is very important because each word is a keyword and is an instruction, basically. It's a statement for the computer. So it's very much different than a natural language, which is very, very ambiguous. Also, the computer programming language should be writable by a human, and in a way that is why it's called the high level, because it's closer to English, which is a human language. I'm not for the English for the language of the world. I don't want to go into this discussion, but definitely which we need to choose one example of a human language, which is English. And then we build up something which can understand those keywords, which are a subset of English, okay? But also it should be readable by human later on, after months, after years, which it should be easily maintainable over time, all right? Because this has happened to me when I was younger, mostly, that you write a program nowadays, everything is clear, everything works, everything is perfect. Then you put away in a hard drive and you go back 1 month later and you go and read it, you don't understand nothing about it. It's like being written by somebody else. And this is very bad from a software engineering point of view, because you have to write in a way that is maintainable. And also language, the programming language that it's also helping with the syntax in order to be maintainable. And this is one interesting aspect. Why not also to express abstract data representation without requiring too much care of digital memory handling? Because I don't want to go into details of pointer, I don't want to go to see how much memory my computer has, I don't want to see how many computer cores I have, I don't want to see how large is the hard drive. Well, mostly these days, because it's like basically digital memory is cheap, it's almost free, and the days of a few kilobytes to make your embedded computer to work, or the Commodore VIC-20, the first computer I used in the 80s, they only had 3 kilobytes of memory. So that was another era of computer science, right? Also, it needs to be expandable, because with a language I can do something, but then I want to build up more new functions, new libraries, new language extensions, and so this should be able to do it easily as well. Also, I wanted to be agnostic towards a particular programming paradigm. We will see that later, but it's not possible all the time, because there are many programming paradigms. We will see that Python as a language is covering most of them, okay? It also has to be agnostic towards the operating system. I don't want to write a program that runs only on this Windows 10 machine, right? And then when Microsoft decides to go to Windows 11, it doesn't work anymore, right? It's not possible, because I have to do science. I don't want to waste time doing porting of software between operating systems. And also needs to be concise and support compact instruction statements, because I don't have time to write long instruction statements. I want to be short, compact, and good to achieve the goal, okay? So if I'm selling well Python to you, then we can start going more in deep. Where is Python? Python is described in this very nice Wikipedia article in English. You see, it's a multi-paradigm. It's an object-oriented, procedural, imperative, functional, structured, and reflective language. Okay. In computer science, most of the time, these properties are only for one language at a time, but Python actually covers all of them. Has been invented by Guido van Rossum, this guy here, which is now working after retirement at Microsoft, leading the Python team in Microsoft. And it's a 30 years old language, so there is quite a large community in the world. It's open source, it's run by the Python Software Foundation, which is a not-for-free organization that can help programmers and fund the development of the language. Now is 3.95, which has been released on May 2021. Okay, why Python as a programming language? Well, I don't know. It's an interesting study. This is a video reporting statistical analysis of surveys among programmers. And if you start from 91, the most used language was C, which is right one step over machine language, but still very powerful. You can do everything with C. Actually, Python is written in C, and thanks to C, Python exists, okay? So C is very important for an engineer, for somebody who's graduating in computer science, as well as all the C-derived languages like C++, C#, Java, and other things. But then after 1999, you can start looking at the bottom of the scale. You see Python here, it's very small blue one. At the 2004 is still on the lower bound, and Java is the winner at the top. And look at this. At the third quarter of 2019, Python is the most used programming language among programmers in the world. So I don't know if I can ask this question, if I can answer this question, but definitely there should be a reason why people is attracted by this language. Also, non-computer scientists are attracted to this language. Okay. Also, look at this. What is the usage of Python in which fields and which disciplines? Well, just looking at computer science itself, the most usage of Python is in 2018 survey was 58% in that data analysis. So data science, the so-called new data science. At DSIM, we actually have a master's degree in applied data science. And on next Saturday, we're going to have the first graduation day for data science. So you're welcome. And but anyway, so you can understand that data analysis is basic for science. That's why I want to sell Python for science, because it's, you can do very good data analysis with Python. There are many, many other things. Web development, machine learning, software testing, education, software prototyping, desktop development, computer graphics, which is less and less because there are other languages that are more appropriate to do those kind of things and applications. Okay, so it's not a language for everything. Today we're talking about Python as a language for science. Well, why we do want to do that? Well, let's have a quick look at this problem. Okay, we express this to find the solution of a second grade algebraic equation. Here we write down the equation ax squared plus bx plus c equals 0. You know the well-known formulas to extract the solution. Consider that this sqrt is a square root function, which it cannot be calculated for negative numbers. So you need to be careful about this quantity, the discriminant, if I'm not wrong, which needs to be more than or equal to 0. And also this parameter a should not be equal to 0. Otherwise, you cannot do division here, right? So this small program, which has only two functions, one is not related to this problem, which was about text splitting a sentence into words. It's in Italian, but you can follow it. This is the main program. Okay, you just input values from the user, A, B, and C, and then you call a function which is called soluzioni dell'equazione, where you just calculate every part of it, the discriminant. If it's more than 0, then if A is more different than 0, you can return solutions. You can definitely build a better function than this, but I want to just show you how short is the program to make to kind of give a first prototype as a solution of this equation. Okay, but why Python for science? Again, I don't know if I'm the right person to answer this question, but definitely Nature judged it to be worth publishing this interesting paper, which is a scientific Python fundamental algorithms for scientific computing in Python, which appeared a few years ago, I think in 2020, yes, February 2020. And this is the link to go to Scientific Python website, where the very important SciPy library has been developed, including many, many other libraries, such as numerical Python, where they are accelerated libraries which are using other languages like C, Fortran, and many more high performance compiled languages, to give to the Python environment a plethora of all kinds of functions you can do in science. I reported, this is the history as reported in the paper. Started in 2001. There were many, many steps, adopting numerical Python in 2005, for example, using a compiler, Cython, in 2007, and using BLAS, which is a well-known Fortran-based library for linear algebra, optimization included. The very first version being developed after 16 years, okay? So when I heard about the European Union, they want us to make a result in 3 years. Okay, well, look at this and see if you can publish on Nature after 16 years, okay? Consider this. What kind of sub-packages and libraries can we see on scientific Python? Look at this, many. The Fourier transform for the signal analysis, physical constants, integration, numerical integration, interpolation, data input and output, linear algebra, whatever, signal processing, image processing, linear programming, sparse matrices, and many more. And I just pointed out this red one, statistical function, which itself has a subset of many functions. So you can tell that you already done some of this in your laboratory, right? To analyze statistics of your experiments, to do simulation, to analyze the results versus control, and this kind of stuff. You can also do that, everything in scientific Python. Then, why not we get our hands on Python? How we do that? Well, very easily. You can even use Python in Excel. This is a commercial product that allows you to do small Python programs inside your Excel spreadsheet. I disclaim that I do not endorse this commercial product and any other commercial product I'm talking about in this presentation, but this is useful for a scientist, right? Also, I have a question for you. What is big data? Well, my answer is everything that doesn't fit as an Excel spreadsheet, okay? A lot of scientists try to make science with an Excel. Well, I understand to start with, but then as soon as your data reaches millions of rows, you cannot use Excel anymore, right? So please switch to Python and scientific Python, because you can really do big data, gigabytes of data analysis, you name it, without limitation, only given by your underlying hardware, physical memory. You can get Python from nowadays from the Microsoft Store very easily, and instead of installing video games, why not install Python and start to play with it, so you can learn by yourself. And also you can, you don't want to install anything, right? Then you can use an online. Okay, this is a Python idle, which is the right you get from Microsoft Store, or in macOS is already included as a Python interpreter. But if you don't want to install anything, you can use an online Python programming environment on repl.it, which is read, evaluate, and print it cycle. You do everything on a browser. So you can develop your next experiment online without installing anything. Just maybe from your smartphone. You can actually install Python on a smartphone, okay? It's called the QPython. And also on your tablet, on your iPad, whatever. Now, let's get back to science. Well, look at this. This is just some Python extension for science. And you can imagine, has so many software projects and so many researchers behind all of this stuff. Python is the core, and then you have Jupyter Notebook, very important for a data scientist. Numerical Python, you can forget about MATLAB and things like that. Pandas for big data analysis, Matplotlib for graphical visualization, as well as Bokeh library, and SciPy, the one I'm talking about before, and then all the scikit extensions for image processing specialized, SymPy for symbolic Python calculation, so instead of Mathematica, for example, and then AstroPy for astrophysics, BioPython for biology. Another thing, there is also a GeoPython for geosciences and earth sciences. So why don't we go and look into some scientific disciplines now and see how Python has been used over the years. I'm going to go faster, so I'm sorry because I wasted time at the beginning. This is my own research field, which is cognitive robotics. I published this mind map in 2018 at the European Scientific Python Conference. And each one of these leaves is a Python library. Some of them have been developed by my laboratory, my students, myself, like PyDali, other like PyIML I've been working on. But there are many other libraries like PyTorch and TensorFlow that are made by Google and Facebook. Scikit-learn from a French research institute. And then all the robots that I have in my laboratory, you can have a look at those two rovers. They are all Python based, and my students can easily get started on doing robotics thanks to Python and computer vision in real time. But forget about what I do. Let's go and have a look at other things. How about this? Okay, I never understood quantum physics and more importantly, Schrodinger equation. These are the requirements to run this simulation that you can get from GitHub. You can easily install on your computer. It's requiring numerical Python, matplotlib, which is for graphics, and scientific Python for all the integration and partial equation calculation, and a 3D graphics, and to compute eigenstates, okay, for quantum physics, which is H5Py language library. And you can actually get this kind of animation with this complex calculations and visualization. So this is energy level of some quantum state, and you can see how the quantum matter actually is making us to have fun and trying to understand for the first time what is Schrodinger equation about. Still quite dark for me. Well, look at this, the photograph of a black hole. I've been honored to meet Dr. Sarah Yasson at our conference, European Scientific Python 2019 online, unfortunately. But she was one of the author of this work that was published on Astrophysical Journal Letter. And basically, they used Python to do all the data collection from Earth-based horizon telescope, measuring many radio signals from different sites, and fusing all the data in a single source, and then doing inverse convolution to calculate the possible image of the black hole, indirectly measuring the gravitational distortion of space and light around it. Okay, so you can see here these are the years of the research as the picture was actually making better and also animated, because the black hole is very fastly rotating. I put here a link to her presentation at our conference. You can have a look here. I don't have time to show you, but definitely you can understand here how Python was useful and was been used to make the data representation and the visualization of big data with Python. Okay. Another example, we can have a look at the BioPython. To have a look at this, this is an example of a virus, Zaire Ebola virus glycoprotein structure we visualize from the protein data bank and using BioPython library. You can also do many other things like genetic research with BioPython. This is a presentation that I linked in the slides. You can have a look at this, and it's very illustrative, and also tell you the history of Python and how this you can actually apply to sequence analysis and things. So you're welcome to go and have a look at this at your will. Now let's go back to my presentation. Okay. So again, you have a link to this PDF here. Then another application in chemistry, an interesting talk. You want drugs? Use Python, okay? Because you can actually do drug discovery with Python. And from Michael Nowata, and again at the European Scientific Python 2015 presentation, where he described how Python is leveraged to streamline the modeling of drug discovery data and the development of tools for the scientific community. Various examples, chemistry toolkits, machine learning application, and web framework to show how Python can glue it all together to create efficient data science pipelines. So this is again a getting started scientific Python specialized in chemistry and use it for drug discovery. Two more last examples. So this is neuroscience, again presented by Santiago Villalba at the European Scientific Python 2015, and it's reverse engineering animal vision with virtual reality. Present Python tools for analyzing large collection of stimulus response time series, which are taken with electrodes from the brain. The PyData ecosystem provides proficient at all levels of data processing pipeline, from collection to discriminative analysis and interpretation, and so on. We introduced new libraries to do this work, and so please have a look at this. And also, that's it. I finished my list of examples because it would be never ending. I want to just leave you a message: unite and debug the world with Python for science. Okay, that's all. Thank you very much for the attention. And if there are any questions, I will be happy to answer.
Okay, thank you so much, Giovanni. Thank you for your interesting presentation. Now, are there any questions? Yeah, there is a question from Michela Ciocca. She says, what do you think about R Studios? What are the differences compared to Python? I suppose he's talking about language R.
Yes.
Considered the competitor in data science.
Definitely, it's a complementary tool. A data scientist should learn both, okay? Because R is a language specialized and developed by statisticians and people coming from a statistics background, and it's a very, very well curated libraries for statistical analysis, statistical learning, and data analysis. I have to say that Python is more general. So you can have a similar set of functions. Probably like 85–90% of coverage of what you can find on R. But then what you can do with Python is to integrate with other tools, with the other things that may not be available on R. For example, you can, for example, integrate with virtual reality, or you can integrate with a web development environment, or you can integrate with a robot, right? So that's why, as a computer scientist and engineer, I do prefer Python, and I adopted Python for my robotics course. But definitely, indeed, in data science, applied data science master degree, we teach R and Python. Okay, so you cannot actually avoid R. More questions? I see the question. What is the role of Python in network programmability? I also can ask to Ivan because he's an expert on networking. Definitely, there are many libraries for network communication, protocols, all kinds of back-end services, databases.
Yes. It's very useful, especially related to cybersecurity, network cybersecurity, network penetration testing. very useful script. You can write a useful and short script to evaluate, for example, traffic in a network and so on.
Also consider that network is quite an ambiguous term. You can use in computer networking or, for example, social networks. And again, you have a lot of tools with Python to do social science studies in social networks. You can also do network analysis on data. And again, there are many libraries and functions on scientific Python tool to do that. So it definitely needs to, we need to understand what's the understanding of networking that you're talking about. Any questions?
Okay. Some other questions seems no, but if you have few seconds, I have a question for you, Joe. There is another question from Marco Ponsetti. And how can we trust the analysis of big data, given that the brain of a human being is not able to handle such a large amount of information and cannot control if there are mistakes? I hope you have understood.
Yeah, I may not have an answer. I'm telling you. Well, but definitely, since the scientific Python library set is kind of certified, right? It's peer reviewed, certified by community of scientists who have been applying those libraries for data analysis in many scientific fields. It's like, first of all, you cannot certify software. That's a theoretical aspect. There are people that are doing research in how to certify the correctness of an algorithm. It's an open research field. I have to say that by experience, by experimental results, definitely those libraries are good enough. Think about that the period to stabilize scientific Python was 16 years. And that was 16 years of peer reviewing code among computer scientists, physicists, biologists, and all scientists that were using the library in their own daily experiments. So I cannot answer this question, you know. Definitely, we are on the shoulder of giants. So if you start tomorrow to use scientific Python, you are using the experience of I don't know how many thousands of scientists and researchers, computer scientists that built that background. Okay, so we can trust them. Thank you.
Let me clarify. The question was from
no, Marco. If there are any more questions,
not from me. Can you hear me? Hello, can you hear me?
Yes. Okay, I switched off my speakers before. Can you repeat, please?
There is a question from someone. I heard something, but if he can repeat, now we can. Okay. There is Mauro Maccarone.
The question was from Professor Anateti, not from Marco Ponsetti. I cannot take the credit.
Okay. Yes, the question was from Anna. Thank you, Anna.
The question about big data, I suppose, and if it's trustable, I suppose the training of, for example, a neural network with this big data. But I'd like to add something. There are a lot of strategy for validation, the training of a machine learning model, for example, neural network. But there is, as said Giovanni, there is not an answer, a perfect answer. And now, since there is Antonio Maurizi, maybe he want to say something. I don't know, or is the next speaker? So I think that we can close. Thank you again, Giovanni.
Thank you.
Thank you, everyone.
Okay, I think we can go on with the biology session. Can you hear me?
I do, if it's me to answer.
Okay,
Yes,
okay, that's perfect. Okay, welcome to the biology session of the TMS Day of Science. This is the last session of the day. We will have a plenary lecture plus 2 selected oral presentation. We can start with the plenary lecture, and it's my great pleasure to introduce Professor Mauro Maccarone. Just few words about Professor Maccarone. He is a full professor of biochemistry here at the University of L'Aquila. He got his PhD in enzymology and applied to medical science here at the University of L'Aquila in collaboration with the University of Tor Vergata in Rome. Then he spent some time abroad in Netherlands at the University of Utrecht, where he was a young researcher and then a researcher. He's member of the editorial board of scientific journal, and he's also a referee of many of these journals and for conference. He received many awards. I would like to mention the last one in the 2020, the UU award for his work on the endocannabinoids. He was also a chair of a famous conference, so the Gordon Research Conference on cannabinoids function in CNS. Some numbers about Professor Maccarone. He published more or less 500 papers. He has 18,000 citations with the NH index of 70, and he has been granted by many founding agencies, national and international agencies, and he also received grants from the European Commission. So the lecture of today is entitled Lipid Signaling in Space Biomedicine. And so, Professor, the stage is yours.
Thank you so much, and Dr. Maurizi, Antonio. And so, should I keep approximately half an hour for this presentation, or should I shorten a little bit?
If you short a little bit, this way we will have time for an active discussion, so 20-25 minutes is fine. So we will have time for discussion.
Let's see if I can share the screen, because we start. Can you see my screen?
Yes.
Excellent. So thanks to the organizers and to Anna Maria Teti in particular for this nice opportunity to share with you some information on a topic that somehow is a niche, but I chose it on purpose just to stimulate young participants to look ahead and maybe even more than usually they do in the labs. So I thought to share with you some information on lipid signaling in space and how this might impact on biomedicine. Why? Because historically we are interested in lipids, the biochemistry of lipids, and we know how complex the metabolism behind lipids is, not only in strict terms, so enzymes that biosynthesize and degrade lipids, but also for the signaling that is triggered by these compounds. So looking at the many receptors, here we have an example for the first category I will discuss with you. So many receptors that have to translate the signal brought about by the lipids in cellular responses. And there is also a last reason that I will show in the next slide for being so interested in lipids as cellular signals. I will briefly touch three main categories of lipids. The first one is the eicosonoids, then there will be the endocannabinoids, and finally the specialized pro-resolving mediators. Eicosanoids are, I believe, very well known because they have been disclosed as key modulators of a number of cellular effects, both in physiology and pathology for humans, years ago, to such an extent that it was in the 80s that a Nobel Prize was awarded to Sir Samuelson for discovering the first of these compounds. They all come from the arachidonic acid, here in the middle of the slide, and this fatty acid is able to generate via specific enzymes called lipoxygenases, cyclooxygenases, and we might also add cytochrome P450, can generate different compounds with a very specific biological activity on their own. And here I ask you to simply notice that one of the enzymes of the eicosanoid metabolism, so-called 5-lipoxygenase, is actually quite pivotal for the synthesis of both lipoxins, compounds with activities in immunity, and leukotrienes that have many goals in our body. Just think of asthma. Why 5 LOX? Because this was the first practical example that we could investigate under microgravity conditions. And the reason why we were interested in this enzyme is because it represents a very relevant for biology, as I said, but also very peculiar catalyst able to use a lipophilic substrate like arachidonic acid to build up more hydrophobic products. So enzymes working with lipids are a unique opportunity to investigate interfacial catalysts. And this type of catalyst might be really engaged by phenomena like microgravity, where, for instance, the distribution, we should say the dispersion of the lipids in the surrounding watery milieu might be favored by hypogravity and disfavored by hypergravity. If I can make a very simple example, just think of the drops of oil that you can put on water, and if you use a centrifuge at very low acceleration values, you will separate immediately the lipid part from the hydrophilic part. The opposite might occur when you instead reduce the force of gravity. The fact is that when we performed a real experiment under microgravity conditions in what is called the parabolic flight campaign, allowing at least the time of a catalyst of a true authentic microgravity, we could measure the catalytic constant of the enzyme that are the Michaelis-Menten constant, Km, and the maximum velocity, the Vmax. Of course, I don't want to bore you with basic biochemistry, but let's say these are the parameters that identify the ability of a catalyst, of an enzyme, to work on the substrate. What we found is that the overall efficiency of lipoxygenase was improved in space. And this was basically due to a better ability of the enzyme to find and then degrade its substrate in microgravity, in line with the idea that more molecules of substrate can be available to the enzyme because they are better dispersed in water in microgravity than in normal. So we had the first clue that a specific molecule, an enzyme working on lipids, could be indeed a gravity sensor, a target for microgravity. And building on that, we set another experiment, this time flown on the International Space Station after approval by the by NASA, the ESA, European Space Agency, and the Italian Space Agency, where we wondered whether a enzyme could also be involved in phenomena like programmed cell death that were known to occur in human lymphocytes in microgravity, with an impact on the ability of astronauts to respond to exogenous stimuli. So what I mean is that for ages, we know that under microgravity, the immune system is somehow depressed, but nobody understood why. And we simply contributed to shed some light by showing that microgravity, by activation of lipoxygenase activity, could improve a chain of events, ultimately leading to programmed cell death. So, reducing the number of immune cells in space and therefore reducing the ability of astronauts to respond and to have an immune response in that environment. Of course, the implications of these are many, and the countermeasures that must be taken are essential if we want to really explore and finally conquer any planet outside Earth. But for now, we had a clear information. Microgravity can affect enzymes and can affect metabolites, eicosanoids involved in programmed cell death of immune cells. We then continued our interest in this field by investigating a different type of bioactive lipids, again built of arachidonic acid, but this time with a little addition to the fatty acid, that is an ethanolamine or a glycerol moiety that generate respectively an arachidonyl ethanolamine or anandamide for short, or 2-arachidonyl glycerol or 2-AG for short. Again, you see that we have a number of metabolic enzymes that can build anandamide or 2-AG, different enzymes for each, and then a number of enzymes that can degrade both compounds, with, again, a number of targets, receptor targets, that are different from those activated by the eicosanoids. And to have a kind of a short glance at the ensemble of proteins, lipids, receptors, enzymes that all together form the endocannabinoid system, here you can have this view where you see that anandamide, like 2-AG, can activate the cannabinoid receptors, CD1 and CD2, G protein coupled receptors sitting on the cell membrane with the active site outside, or they can activate the vanilloid receptor, this time a channeled receptor, not a G protein coupled receptor, with the active site inside the cell. There are many enzymes, as we have seen, for anandamide and 2-AG, but for the sake of this presentation, please keep in mind these two cannabinoid receptors, CD1 and CD2, activated by anandamide, and the metabolic enzymes for anandamide, a phospholipase D called NAPE-PLD, and that makes anandamide by releasing it from phospholipid precursors, and an hydrolase called FAAH that is able to cleave the enzyme. And this is why the reason is that we will show you data on the alteration of these specific molecules in microgravity. The other reason why I asked you to focus on those molecules is that it has been known for a long time on Earth, especially thanks to the seminal work of the late Itai Bab, a friend, a real expert in this field that passed away a few years ago, that type 2 cannabinoid receptor is critical to achieve a regular bond formation with a proper trabeculae that even when you only miss that receptor in a knockout animal, are much less strong, much less formed, and therefore weaker preluding or contributing to osteoporosis. Or in general, we and others could recapitulate that from type 1 cannabinoid receptor, CD1, we have a number of actions on the tissues that in general are in favor of osteogenesis, so they favor the making of bones. Whereas type 2 cannabinoid receptor can contribute to trabecular formation, but it can also have a number of activities at different levels that altogether stimulate osteoclasts, so bone degradation rather than formation. At any rate, these two types of receptors are known on Earth to be quite critical for bone formation. And I had this issue of osteogenesis because it was one of the topics that we addressed in one of the last space missions involving also the endocannabinoids. And to make this clear, or clearer, I also like to add another piece of information, that is endocannabinoid system, and again CD1 and CD2 play a clear role also in the formation of osteoblasts and osteoclasts from stem cells. You know that from the mesoderm you have both hematopoietic stem cells and mesenchymal stem cells. It is clear, again from a number of Earth experiments, that CD1 favors osteoblast formation and therefore bone formation from mesenchymal stem cells, but also from hematopoietic stem cells. Just look at this graph, and we of course don't want to go into too many details. On the other hand, CB2 might play a less clear role, exactly as it happens with tissues, because it can both stimulate osteoblast formation, but also stimulate osteoclast formation, leading to bone resorption. In particular, you will see that from hematopoietic stem cells, the role of CB2 appears more against osteogenesis than in favor of it. So, having all this in mind, we started to investigate a possible alteration of endocannabinoid metabolism in space, again flying a second experiment on the International Space Station, the so-called Roll 2. The first one on the immune cells was rolled. And here we found a clear imbalance of anandamide metabolism in the sense that a reduced degradation of anandamide, reduced FA activity, and an increased biosynthesis of anandamide through an increased NAPE-PLD activity. The latter is the biosynthetic enzyme. The first is the hydrolytic enzyme, as I mentioned before. Overall led to a higher tone of anandamide and then expectedly a higher activation of its target receptors. We then started from that information to design a new experiment on endocannabinoids, the third project that we were able to fly on the International Space Station, where we did something different. Instead of using immune cells, we took again from peripheral blood a portion of immune cells, the peripheral blood mononuclear cells, from which this time we deprogrammed some of the cells to achieve blood-derived stem cells. And these cells were the object of the experiment because we found ways to drive these blood-derived stem cells towards an osteogenic differentiation via treatment with rapamycin and the stimulating scaffold. This osteogenic differentiation was the subject of our investigation, where we interrogated the endocannabinoid system, of course, but also additional embryonal or differentiation markers to see whether from blood cells re-educated, reprogrammed to become osteoblasts, we might help the synthesis of bones that could be helpful for the astronaut to compensate osteoporosis. The basic idea was that the same reprogrammed cells could be re-injected to the astronaut from which we had taken blood. And this is the model. We know how rapamycin plus a certain scaffold can drive differentiation of blood-derived stem cells into true osteoblasts. And this experiment that belonged to the Biomission Vita, the Italian Space Agency, was made possible in space also thanks to a specific hardware that we needed and that was developed on purpose by Kaiser Italia. In essence, what we did was to send in space blood-derived stem cells. Then, when in microgravity we injected the inducer of differentiation towards blastocysts, that is rapamycin and the scaffold, and at the end we blocked everything with a fixative that allowed this material to remain in standby, frozen, and then delivered to the lab on Earth for further analysis. And this is a general strategy, just to inform those of you who might have less experience with space research. You have to set pre-flight experiments that can be feasible on Earth. Then you load your experiment on board a rocket that will bring it to the International Space Station. In our case, it was an instrument called Kubik. And then in space, you must perform your experiment. You must fix and froze in the Melfi your results. And this will be finally delivered to your lab again in the post-flight. So when you have an experiment that you can design according to these constraints imposed by the mission, you can finally have your data exactly as you do with the normal experiments. And what we found is that Fa, the hydrolase of anandamide, was certainly reduced in space compared to Earth 1G controls. CD1 instead was increased in microgravity, and please remember that this is the good receptor, the one favoring osteogenesis. Instead, CB2, that is the bad, or at least the unclear receptor, certainly bad when you start from hematopoietic cells, as we did, CB2 is decreased in microgravity. So overall, because none of the other elements of the endocannabinoid system that we tested was either expressed or affected in microgravity, what we could conclude is that overall, in these blood-derived mononuclear cells, we had an increase of anandamide levels due to reduced degradation, and therefore the possibility to overstimulate, further stimulate CB1 that was also overexpressed, whereas CB2 was reduced in space. Is anandamide a pro-osteogenic factor via CB1? This can well be based on the data that we know from Earth literature, as I showed you at the beginning. And what we are investigating now is to what extent anandamide can also contribute to a change in key modulators of osteogenesis, change that we did observe in space. And in particular, we could demonstrate that SOX17 and GATA4 that are normally along the pathway that from commitment leads stem cells through differentiation to osteoblast formation, these two markers are indeed activated by microgravity more than they are on Earth. And in keeping with this, consistently, if you want, key repressors of osteogenesis like snail and OTX2 are instead inhibited in space, opening up gene transcription that is needed to convert blood-derived stem cells into osteoblasts. overall, what we can say, and we publish, so for any detail, please look at this paper that is free on the PubMed. We found that it is indeed possible to take blood cells from someone, they program these cells towards an osteoblastic lineage, and via endocannabinoid signaling have reached formation of osteoblasts, therefore more bone formation. And in microgravity, this happens better than it does on Earth, opening also the possibility to take advantage of this lipid signaling pathway for further therapeutic attempts that could be helpful also on Earth. And I like also to remark another point. We have used blood-derived stem cells, so the hematopoietic lineage, which might be more relevant in the light of recent data from another group, showing that instead the mesenchymal stem cells do not seem very responsive to osteogenesis, to osteoblast formation, because with this pathway going through TAZ, in space, not much of activation was observed by them. In other words, pointing to hematopoietic cells as possibly more promising than mesenchymal stem cells if you want to improve osteoblast formation. So that was for the endocannabinoids and previously for the eicosanoids. What about a third family of bioactive lipids, like the so-called, collectively called, specialized pro-resolving mediators, SPMs for short? Also, these compounds derive from fatty acid, like arachidonic acid, as the first two families, but also additional fatty acid with even more double bonds, like eicosapentaenoic acid, EPA, or docosahexaenoic acid, 6 double bonds, DHA. Of course, arachidonic acid has only 4, eicosatetraenoic acid. From these polyunsaturated fatty acids, a number of derivatives can be made now by different enzymes that can be partly shared with the eicosanoids, like lipoxygenases and cyclooxygenase 2, but under specific conditions. For instance, when you have the possibility to activate, to modify with acetyl salicylic acid, aspirin, these enzymes. So just to say, again, we have a lot of complexity in the metabolism, but this metabolism seems specific for these compounds that, unsurprisingly, have a number of new receptors, most often G protein coupled receptors, that are different from those activated by eicosanoids and those activated by endocannabinoids. What we know is that these mediators, STMS, are fundamental for a process that is called resolution of inflammation. And this is a key in so many pathologies because it is the reason why an inflammation, either chronic or acute, that is used by the organism to respond to an insult, an exogenous insult, like pathogens or others. This inflammation at a certain point is stopped, because if you don't stop it, it becomes the source of a big problem that leads to a number of diseases like cancer and many others. So the ability to block an inflammation that has been triggered to protect the organism, and this blockage of inflammation is called resolution, is equally important as inflammation. If you don't have a proper resolution, you don't have an homeostasis, and you have problems instead of solutions for our health. And the point was that although many active lipids and molecules have been investigated in space, by us and others, and I've just shown you, if you want, our personal contribution to the field, never SPMS have been investigated so far. And so we have running experiments, ongoing experiments, where we are wondering if these resolvins or maresins or protectins, as the specialized pro-resolving mediators are called, can be in fact modified by microgravity. And to do this, we are now running a setup, the following setup, again starting from human peripheral blood and isolating peripheral blood mononuclear cells, immune cells. We do artificial microgravity with the rotary cell culture system in the lab, and then we perform quantitative analysis of gene expression and on production of these lipids. Here I'm also almost at the end to share with you preliminary data clearly showing that microgravity, but this time we have simulated microgravity, and we hope to have an access to a next campaign on the ISS, but clearly microgravity can increase the expression of some key receptors like ALKs or ChemR23 that are targets of resolvins, or GPR18. As you see, at zero gravity, microgravity, expressions are much higher than at 1G. And also one of the mediators of resolution called resolvin D1 is modulated in its expression, this time with a decrease, under microgravity. So microgravity seems to be actually a player for lipid signals, seems to be something that lipids sense maybe better than other molecules. And once again, maybe this interfacial activity of these compounds that have to be in water, but they don't like to be there because they are lipophilic, could be a key to interpret a number of conditions that are known to occur in microgravity. And I focused, as you have seen, on the immune depression and on osteoporosis. Our work, of course, was done thanks to many collaborators, but here I like to thank my group and people from Kaiser Italia that have been really next to us for all the missions to space. And in particular, I like to thank Paolo Nespoli, or Astro Paolo, who was the one to actually perform our last experiments in space in the context of the Vita mission of the Italian Space Agency. I think it took a little bit less of my time, but I hope that the message reached you in a clear manner, and glad to take any question you might have. Thank you.
So thank you, Professor Maccaro. Yeah, you were very clear, and it was very nice talk and lecture. So we have some questions. We can start with the ones from the chat. So the first one is from Marco Ponzetti. So he asked, based on the differentiation ability, I assume that the BDSCs are similar to the mesenchymal stem cells for the purpose you are proposing. So what about using the urine-derived stem cell instead? They would probably be easier to obtain with the lower risk involved.
Well, thanks, Marco, for your question. Actually, I don't know specific in detail the specifics of these urine-derived stem cells, but certainly if you can prepare them and grow them in a way that is compatible with the constraints of a space experiment, and this unfortunately is very often the barrier to a certain activity in space, because you cannot do it, and astronauts cannot spend too long time on your experiment because everything is very restricted. But if you can do that, I think there is no reason why it shouldn't work. So it's something that should be tested.
Okay, yeah. Then we have another question from Michela Ciocca. So she asked, can microgravity also affect mature osteoblasts?
Again, in principle it may, but when you want a clear-cut answer, you have to do the experiment. And again, the question is, are you able to keep mature osteoblasts in a culture system that can resist the constraints of a space experiments? I know that this is a no answer, but certainly you cannot have one unless you do the experiment. But just to point out how relevant it is to have the hardware and, if you want, the micro lab available for your experiment, because this is as important as the scientific question. It must be feasible, safe, and all things that space needs.
Okay, so I have one question, since there is no other question in the chat. So you mentioned that when you do your experiment in microgravity on space on the cells isolated from blood, you have an increased expression of the CB1 that has an osteogenic potency, but it's known that the absence of gravity, so the skeletal unloading, induce a loss of bone mass. So do you think that these circulating cells maintain or has the potency to really make bones? So they're able to go in the bone tissue and make bones?
This is an excellent question. What I can tell you is that the idea, the strategy of taking blood cells and retransfusing in an autologous way, re-educated, reprogrammed cells is feasible. There are a number of therapeutic interventions where you do have a blood retransfusion, autotransfusion. So it is feasible, but we do not know how this will translate into a real benefit, because we could only perform experiments on the cells, human cells, primary cells, but nothing on the astronauts. That would be, hopefully, the next step, and I must say could be done also on Earth. But there you need a clinical trial that is not too easy to set. But we would love to know, okay, we now do this also to patients on Earth with osteoporosis. Actually, it's not even a disease, it's just a physiological condition. What benefit do the patients have?
Yeah, this one was my next question, because also on Earth we have a condition of skeletal loading, also the bed rest, the long-term bed rest. So when you fracture or people disease, or if we think now to COVID patients that they stay in the very severe cases for a long time in the bed. So yeah, this was my next question. And the very last one, technical question. So when you use the rotating vessel, of course, it's not exactly 0G like for the astronauts, and there you have also shear stress because you have a free fall of your cells into. Are you? And this is important for both. So your cells just scratch, and the loading on the cells, and also the inflammation, the release of inflammatory cytokines, it could be different compared to microgravity. Are you taking into account also this aspect, or you're just waiting for the next space mission?
Well, these are also very good points. You know, we have been using this rotary cell cultivation system that was developed by NASA for many years now, and usually we have also always compared data with the true microgravity conditions. And for now, we consider it a preliminary information that, of course, without a real campaign on the ISS or other possibilities, but with true microgravity, we cannot address. I know, I can tell you that under our conditions, we reach something like 10 up to the -4 G, which is not bad. And we try to compensate due to the experience with the density of the system, the relevant problem of the shear force. But again, is a preliminary approach, hoping to have access to real microgravity and see what happens there.
Yeah, yeah. So I was asking because also in our lab we have this kind of apparatus, and we face always with this problem. And
I know that Professor Teti and of course all her collaborators have a very long and outstanding experience in this, which is a niche if you want, but with so many open questions and answers needed, not only for space, but like for immune depression or osteogenesis, a potential for therapy on Earth that we desperately need, because there is what is available is not always available for everybody. Just think of some of the therapeutic agents used for osteogenesis, very heavy to use, but you know.
Yeah, and now we have to consider that a new era is starting with the tourist space flights. So, like the first one of, so the problem will become serious. So, hopefully in the next years. So
absolutely, and we could have ways to get answers to those questions.
Yeah.
So we hope to be able to do that.
Okay. So, Professor Maccaroni, thanks again for your wonderful talk. And
thank you all.
We can move on with our selected oral presentation. So we can start with the first one that is from Pier Giorgio Patrizzi. Pier Giorgio, are you online?
Yes, Antonio, I'm here.
Okay, so switch on your camera and share the screen. So the title of the presentation is Phenotype of the First Mouse Model of Carpenter Syndrome. So I can see your screen.
Yes, Antonio, just a moment. I have a problem with the camera because I have not the control of the camera.
Okay, just share your screen, and this way we can start with the presentation.
Okay, can you see me now?
Yeah.
Okay, I share my screen. Yes.
Okay.
Can you see my screen, Antonio?
Yeah, yeah. Okay, but Giorgio, you can start.
Yes. Okay. Thank you, Antonio, for your introduction, and thank you to Professor Anna Teti, all the TMS group for this opportunity. How Antonio said, today I will talk about preliminary phenotyping of the first mouse model for the Carpenter syndrome. But what is the Carpenter syndrome? The Carpenter syndrome is a rare genetic disease displaying autosomal inheritance with a prevalence of more or less 1 on 1 million. And to date, this disease has no cure. The Carpenter syndrome affects mainly the bone, which becomes brittle and fractures many times. In fact, affected individuals experience non-traumatic fractures, frontal bossing, ocular proptosis, communicating hydrocephalus, and craniosynostosis. Moreover, Carpenter syndrome patients display skeletal deformities associated with the so-called popcorn epiphysis in the long bones, as you can see here in this image. Genetically, the epsilon 393C heterozygous mutation in the P4HB gene causes the Carpenter syndrome, leading to a tyrosine to cysteine amino acid substitution in the protein chain. The P4HB gene encodes for the prolyl folate oxidase beta subunit, a multifunctional enzyme member of the protein disulfide isomerase family. The PTI is the most abundant protein in the endoplasmic reticulum, and they are involved in the formation of the sulfide bridge in the nascent polypeptide chain, playing a pivotal role in protein folding. Moreover, they are involved in the oxidation of prolyl residues in type 1 procollagen and act as chaperone, inhibiting the misfolded protein aggregation. However, today, no animal models were available for studying the Cole-Carpenter syndrome. So, for this reason, we have generated the first mouse model of the Cole-Carpenter syndrome in collaboration with Polygene, carrying the heterozygous dominant epsilon 393 C mutation in the exon 9 of the P4HB gene, using an O'Key strategy, FRT flanked the neomycin cassette. Here on the left, the diagram of the strategy that we use. And as you can see, we were able to discriminate and visualize on agarose gel the mutant allele due to the presence of the FRT site, as you can see in this image. Moreover, to confirm the results obtained by genotyping, we also perform a sequencing of the PCR product to confirm the presence of the mutation, as you can see here with the AG transition. So, after generation of preliminary Cole-Carpenter syndrome mice, preliminary ROS evaluation of the year regeneration showed no obvious change in body length, along with unchanged body weight, as you can see in this image. Then we decided to analyze the bone phenotype, and micro-CT analysis revealed, as you can see in this image, that the bone volume on overall total volume was significantly and dramatically lower in Cole-Carpenter syndrome mice in red compared to the wild-type litter maze in white. You can also clearly see the results in the 3D reconstruction. In addition, as you can see in these pictures, the reduction of bone volume was associated with a lower number of trabecular, along with an increased separation among them, whereas the trabecular, as you can see here, trabecular thickness was unremarkable. Of note, in line with the fragility of the bone observed in the Cole-Carpenter syndrome patients, the microindentation test, which measures the mechanical strength of the bone, as you can see in this image, revealed that the indentation test and the total indentation test were dramatically increased in Cole-Carpenter syndrome mice, indicating a poor quality of the bone. Then we also perform a histomorphometric analysis that show a significant reduction of the osteoblast surface and the osteoblast number in Cole-Carpenter syndrome mice compared to the wild type. And in line with this, then the gene expression analysis carried out on the tibia of the Cole-Carpenter syndrome mice showed a lower expression of typical osteoblast differentiation marker, including RANKS2, Pol1F1, and osteocalcin compared to the wild type. Finally, very preliminary results obtained from the first Cole-Carpenter syndrome primary osteoblast culture showed a reduction of ALP staining, along with a lower deposition of mineralized matrix in Cole-Carpenter syndrome primary osteoblast compared to the wild type, as you can see in this other image. On the contrary, osteoblast cellular parameters, including osteoblast number and osteoblast surface, did not change in Cole-Carpenter syndrome mice, as you can see here. And in accordance with this, no difference in the serum bone resources on CFX markers was found. So, for this reason, osteoblasts seem to be the most affected bone cells in Cole-Carpenter syndrome. Interestingly, the P4HB gene is widely expressed within the organism, and it has been linked to many other common pathological conditions, tissue-specific and metabolic disease. In fact, data from literature report that the kidneys are one of the organs that express the higher level of the PDI. And based on this information, as preliminary approach, we perform an histological analysis in kidney of Cole-Carpenter syndrome mice, as you can see in this image on the left, and better in this other higher enlargement on the right. The analysis showed that the presence of tubular vaporization, as you can see here, and glomerular shrinkage in the Cole-Carpenter syndrome kidneys with a yellow arrow, compared to the wild type, indicating a possible chronic renal disease. Moreover, it is reported that the PDI acts as a retention factor limiting proinsulin exit from endoplasmic reticulum in pancreatic B cells. In line with this, we found a lower insulin serum in Carpenter syndrome mice compared to the wild type, as you can see in this image, indicating that the mutant PDI affects the insulin secretion, impacting on the energy metabolism. So to summarize, we generated the first animal of Carpenter syndrome, and that for Carpenter syndrome mice showed a severe reduction of bone mass and quality. Moreover, other organs behind the bone are affected by the P4HB epsilon 393C mutation, and finally, preliminary observation revealed a reduction of osteogenic potency of primary Carpenter syndrome osteoblasts. So in conclusion, our preliminary data demonstrate that the animal model of the Carpenter syndrome that we generated in our laboratory represent a useful tool to in-depth investigate this neglected genetic disorder. And the Carpenter syndrome mouse model in the future could be used to set up a therapeutic approach for the disease. So thank you to our funding agencies, and thank you too for your attention and your time.
Okay, so thank you, Pier Giorgio. Thank you for staying in time. So I can start with the one question. So, what kind of therapeutic approach do you think could be useful for treating this kind of disease?
Okay, this is a very good question because we have some strategy that we could use in the future. In particular, we have an idea to use 3 possible strategies. The first is the PDI replacement with the PDI recombination. The second is to use the ER stress and autophagy modulators. And the third, the last, is to use the mutated specific siRNA against the mutated P4HB gene. But we have to improve the experiment in the future. And we select the best therapy.
Okay, so since there is no other question, we are running also a bit late, so we can thanks Pier Giorgio for his presentation, and we can move on with the last speaker of the day, that is Michela Ciocca. And Michela, you can share your screen. The title of the presentation of Michele is Transcriptome Analysis of Not-Too-High Breast Cancer Cells Reveal a New Molecular Determinants for Breast Cancer Cellular Dormancy in the Bone Marrow Microenvironment. So, Michela?
Yes, thank you, Antonio, for the presentation. So, good morning, everyone. Sorry, just a moment. Okay. So, since maybe most of you already know, breast cancer is the second leading cause of cancer death among women. And the major problem is the cancer relapse after years of absence of the disease. That's why the breast cancer cells are able to escape from therapies, and after years, they can generate a new tumor distant from the primary site. Breast cancer cells often home to the bone and may become dormant. And upon reactivation, this small population of dormant breast cancer cells can relapse in bone and in other organs. It's also known that a fifth of breast cancer patients who undergo mastectomy also develop secondary lesion up to 15 years after surgery. That's why now we know that the same population of cells undergoing dormancy should have the adaptability of initiating a new tumor. But who are the actors of this story? We have, of course, the breast cancer cells, and in our laboratory we use the MDA-MB231, that is a human breast cancer cell line, triple negative, so very, very aggressive, and then the osteoblasts. Osteoblasts are, as you maybe know, the bone-forming cells, but here we have a particular type of osteoblast that are spindle-shaped and express high level of encadering. So they are called spindle-shaped encadering high osteoblasts, or SNOWs. But where it happened? So it happens in the bone because the bone contributes to the environment of hematopoietic stem cells, which are maintained in quiescent status by the endosteal niche that is made by spindle-shaped and cadherin-hide osteoblasts. So now we know that a subset of breast cancer cells show an hematopoietic stem cell mimicry and interact with the SNOs to become dormant. In fact, in this recent published article, we already demonstrate the involvement of the Notch2-dependent mechanism in the SNO-induced tumor dormancy. So, based on this, the aim of our work is to use a bioinformatic approach to investigate new Notch2-dependent pathways involved in the SNO-induced breast cancer dormancy. To do this, first of all, we started performing a transcriptome profile of the MDA, Notch2 low and Notch2 high. So basically, starting from the whole population of breast cancer cells, MDA and MB231, we divide them in the ones that express high level or low level of Notch2. Then we extract their RNA, and we perform an RNA deep sequencing in which we found 350 mRNAs that were differentially expressed in the two subpopulations. Then, after this analysis, we decided to perform a bioinformatic analysis using a gene set enrichment analysis on these two subpopulations. So basically, we are able to find the enrichment score of the genes expressed by the cells in a specific signature. So basically, as you can see here in this graph, we are able to find the enrichment score for each gene that is represented by the green line. That is the maximum deviation from 0 for each gene. What we found in this case is that the Notch2 low have an enrichment in the genes involved in the cell cycle signature, and it's also in line with our results in which we found a lower number of cells per field in the Notch2 high compared to the Notch2 low cells, as you can see here by these bars and also by the relative pictures. Then, other way around, we found an enrichment in the genes involved in the pluripotency signature in the Notch2 high population compared to the low, and is also in line with the higher number of oncosphere that we call mammosphere because we are talking about breast cancer in the Notch2 high MDA compared to the Notch2 low. And it means that the Notch2 high MDA have a more stem-like phenotype. Then we go through the pluripotency signature, analyze the hematopoietic stem cell signature, and we found that, again, the Notch2 high cells have an enrichment in the genes involved in the hematopoietic stem cell signature compared to the Notch2 low, and is also in line with the evaluation of the mRNA expression regarding these three genes that are canonical hematopoietic stem cell genes, and we found that the Notch2 high cells have a higher mRNA expression, as you can see here, represented by the green bars, compared to the Notch2 low counterparts, represented here by the blue dotted line. So from this part, we moved in vitro, using the whole population of breast cancer tumor cells, and completing them with a monolayer of osteoblasts, splitting them in SNOs or non-SNOs. And what we found was that the MDA plated on SNOs show the lower proliferation rate after 24, 48, 72 hours compared to the MDA plated on non-SNOs. Then, to better decide the role of Notch2 in the breast cancer SNO interaction, we decided to perform another coplating experiment using the MDA sorted, so separated for the high or low presence of Notch2, and we coplate them with the SNOs. What we found was that the Notch2 high MDA plated on SNOs have the lower proliferation rate over time compared to the Notch2 low MDA completed on SNOs. So we confirmed the role of Notch2 in this interaction. And it's also confirmed by this experiment in which we inhibiting with the genetic inhibition of Notch, or also specific inhibition of Notch2 using a sirna in the MDA plated on SNOs, and it can rescue their ability to proliferate, bringing the latter back to the level of the MDA plated on SNOs, as you can see here. So, in conclusion for this part, we can say that in MDA, Notch2 is a dormancy gene whose effect is potentiated by the interaction with the SNOs. Then, we decided to go back to our RNA deep sequencing to find genes that are specifically expressed by the notch 2 high MDA. To do this, we apply some extrapolation criteria. So first of all, we select the genes known to be involved in pluripotency and HSC mimicry. Then, from this first list of genes, we select the genes that encoding for cell surface protein, so potentially involved in cell-cell interaction, and also for investigated as dormancy genes. From this analysis, we are able to find a list of 10 genes that are expressed by the notch 2 high MDA and in their surface. We validate some of them, and then we perform a network analysis on the protein encoded from these genes. So as you can see here, the red spheres are the markers that we found in notch 2 high MDA, and the other spheres around are the possible ligands of the protein encoded by these genes. Then we select the ones that were expressed in the surface of the cells, so possibly involved in the cell-cell interaction. And at the end, we found that some of these genes are expressed in a higher level in the SNOWs compared to the non-SNOWs. So, in conclusion, the RNA deep sequencing show a differential signature between MDA notch 2 high and notch 2 low, in particular in cell cycle, pluripotency, and hematopoietic stem cell mimicry. We validate the differential signature by conventional methods. We also identify cell surface molecular determinants in the MDA notch 2 high and SNO cells. And finally, we hypothesized that these determinants are likely to be involved in the MDA notch 2 high SNO interaction. So, thank you for your attention, and if you have a question, I'm happy to answer your question.
Okay, so thank you, Michela. So I have a question for you.
Yes, just a moment. Sorry, I cannot. Okay, let's go.
Just, it's just a technical question. How you are going to investigate the list of the interactants that you identify, so to better dissect the interaction between the SNO cells and the breast cancer cells?
Yes, so we plan to silence one by one the proteins, the genes, so at the end the proteins in the MDA and the SNO counterpart, to see if there is some changes in the proliferation rate of the cells when we couplate them. So when we couplate MDA and SNOs, and to better dissect the role of each genes. So this is the plan for now in the in vitro part. And then we will use also for the in vivo to understand if, of course, in the mouse, there is some differences in the tumor develop and also in the metastasis and in the bone lesions, silencing each single gene.
Okay, thank you, Michela. So there's no further question. I want to thank again Michela, and I can invite the other organizer just to switch on their camera, and maybe Marco can announce the results of the poll for the different prize that we have. So Marco.
Okay, so we're waiting for Giovanni as well. Where are you, Giovanni? Professor Teddy is here, Giovanni is here. Okay, I think we are everyone now. Okay, so I'm just going to quickly share my screen so we can announce the 3 winners of today's Day of Science. The winner for the best oral presentation, for the best elevator pitch, and the best poster presentation, or the best poster actually. So let's see. Can you see the screen?
Not yet. Okay. Suspense.
Tell me when you can see it.
Yeah, we can see.
Okay, so many congratulations to the Best Elevator Pitch awardee Francesca Di Donato, the Best Poster awardee Pier Giorgio Patrizzi, and the Best Presentation awardee Michela Ciocca. And of course, an honorable mention for all of the other participants, because it was a very tough choice for us and for the audience. The best elevator pitch award was chosen by you, and we chose the other two awards. Of course, it was a tough choice for us, but so congratulations to all of you, but there can only be one winner. So congratulations to these three awardees for this year. And of course, thanks to Origin Orogen, as they call themselves, for sponsoring these prizes. So you will receive an email soon with the award. Okay, stop sharing the screen. I don't know if Professor Teddy wants to say a few words to close the session. And activate the...
Okay, no, I'm very proud of this TMS, actually. I think it's been really a great event. And yeah, it's nice that we have several disciplines, several expertise, a lot of young people, but also mixed with older people that are more experienced. And yeah, I'm very, very happy. And I think that we went very well all year long. So this is the last event ready for next year. We will have a few changes in the executive committee, and we would like to invite everybody to contribute. There is space for everyone in our working groups, so just let us know, and we will collaborate all together. So thank you very much, and enjoy your holidays.
Thank you.
Thank you. Bye-bye.
Bye-bye. Bye-bye.